y esas cosas.
Pero se va a ver.
Sí, se va a ver, pero prefiero que se vea. Tíralo nomás por el YouTube, ¿cachai?
Ya, sí, lo estoy haciendo.
Y después, cuando ya estemos listas, probar las presentaciones, lo tiramos por Facebook, que es más masivo.
Se va a ver.
¿Ah? Sí, se va a ver, pero prefiero que se vea. Tíralo nomás por el YouTube.
Listo.
¿Me escuchas? Y después, cuando ya estemos. Ay, 4 personas esperando. Ya, espérame, ya. Vuelvo al tiro, ya me voy a ir a sacar el pelo en 2 minutos. nosotros acá. Aló, ¿me escuchan? No, ahora lo estoy hablando por Zoom.
Sí, yo estaba ahí, pero no se veía a nadie.
No, porque pusimos el fondo con la Adriana porque ya estamos transmitiendo por YouTube.
Ya, no, pero...
Hola, Andrew.
Hola.
¿Cómo están?
Bien, bien.
¿Ansioso?
Sí, un poquito.
Sí, va a salir bien. Oye, lamento mucho, hay toda esta confusión anoche porque últimamente pensamos que el keynote speech de hoy día no se nos iba a caer, pero logramos como darle vuelta la mano y igual va a poder participar.
No importa.
¿Eh?
No importa.
No, claro, es solamente por primero había que alargar la presentación, después acortarla, etcétera, etcétera. Bueno, vamos a ver cómo sale.
Consulta, ¿alguien quiere probar su presentación?
Sí, y yo quisiera poner la mía para...
Voy a dejar de compartir, ahora sí.
Hola, Roxana.
Voy a poner algunas diapos nomás, las que tienen imágenes para ver. A ver. Quiero ver una película. Listo, yo estoy lista.
¿Alguien más?
Sí.
Hola.
Hola, doctor.
Hola.
Luis volvió.
¿Ah?
Luis Carvajal.
Sí, sí.
Ya que bueno.
Bueno,
Confusion nomás.
un poco confuso, sí. Yo voy a probar el mío también, Mario José.
Ya.
Hola, buen día.
Hola. Uf, harto eco ahí.
Pero lo callé en la universidad.
Se ve un poco desconfigurado.
¿Ah?
Se ve desconfigurada. Muy desconfigurada.
Es que lo tuve que corregir anoche porque metiéndolo en mi presentación se descuadró todo.
Ah, ya. Pero se ve, por ejemplo, ahí en las líneas de investigación unas no se leen. si lo puede arreglar ahora.
Es
Por
que
ejemplo,
yo
la
lo
de
veo.
la línea 5 sale cortada.
Ah,
La 6 también, es más largo el nombre.
sí. Ahí está mejor, ¿no?
No, es que abajo donde dice cáncer, epidemiología. Abajo de la cara de la doctora Ferrecho sale cortada epidemiología. Eso.
No sé qué pasó ahí, no.
Es que donde achicó el tamaño de la diapositiva se superpuso todo.
No. ¿Dónde está ahí? No sé qué.
Pero ahí se lee.
Por alguna razón sale cortado. Ya, ahí está mejor.
Adriana, por favor, ¿puedes nombrar de coanfitrión a Magdalena Díaz?
Sí, hoy. Listo.
Gracias. Al doctor Cuello, al doctor Corba. Ah, tanto, ya.
Sí, ya está.
Ya, estamos listos.
Hola, hola,
Hola,
Hola.
Vicente.
¿cómo están? Voy a probar solo la página de la portada, es para ver si se proyecta bien o no. ¿Puedo?
Sí, adelante.
Ah, el tiro.
Vicente, hola, buenos días.
¿Hola? Sí.
¿Quieres que te comparta este fondo de... Ay, ya, que no tengo mi pantalla. El de ACCDiS.
A ver. ¿Ahí se ve?
¿Sí? ¿Se ve?
Sí.
Súper.
Adriana,
¿Sí?
Eso
¿lo puedes
quería.
poner en el chat? Porque a mí me gustaría compartirlo también.
Ok, ya, lo pongo.
Ah, el fondo. Entiendo ahora a qué se refiere el fondo.
A esto.
Y voy
Voy
a
a
cambiar la
colocar.
presentación. Sí, sí. Sí, igual
No,
después voy a cambiar de entorno. Es que ahora estoy en la cocina un poquito por el...
no se preocupen. Yo solo si quieren.
Sí, por favor. Ya.
Voy a mandar
Sí,
por
yo lo estaba buscando y no,
Lo encontró.
no. Es que es
no
un
se
link,
preocupen.
es un link a un archivo que está en el computador y cambió.
Y gracias, Adriana. Entonces quedo atento ahí y lo establezco.
Lo vamos a mandar por WhatsApp.
Sí,
Ya,
lo voy a
muchas
mandar.
gracias.
A ver,
Adriana, ¿puedes poner como coanfitrión, por favor, a Leonardo y Daniela?
Voy.
Ariel, ¿quieres probar tu presentación también? Parece que no me escuchan. ¿Me escuchan, cierto?
Sí, la estoy buscando.
Sí,
Ya,
sí,
¿la puedo compartir ahora?
Sí.
A ver. Mm. ¿Se ve?
Sí.
Ya. Estamos bien.
Ok.
Okay.
Andrew, yo quería preguntar cómo va a ser con las preguntas. ¿Va a ser al final de todas las sesiones? ¿Una ronda de preguntas o de cada sesión?
No, va a ser al final ahora, porque vamos a estar un poco justo con el tiempo. Entonces, al final tenemos como unos 15 minutos para todas las preguntas y lo ideal es que la gente lo anoten en el chat. Entonces ahí vamos revisando el chat, o voy yo revisando el chat y voy preguntando.
Ok.
¿Ok?
Ok.
Ya, doctores, vamos a empezar la transmisión ya más masiva. Así que les voy a pedir que por mientras apaguemos la cámara y a las 8 comenzamos, doctor Juez.
¿Tú me vas a mandar un mensaje por chat,
Sí.
María José?
Todos
Ya,
los voy a
ok.
comunicar por interno.
Ok.
Ya.
¿mandaron la foto del logo? ¿O
Sí, le
poner
estoy
otra?
mandando. Lo que pasa es que voy por el grupo.
Ah, ya.
Ya. Voy a apagar las cámaras. Comenzamos.
Okay, good morning, everybody. Welcome to this second ACCDiS Cancer Spring Symposium. My name is Andrew Quest. I am a principal investigator at ACCDiS, the Advanced Centre for Chronic Diseases, and together with Alejandro Corbalan, another principal investigator at this centre, we have put together this program we'll be experiencing in the next 3 days, so November 3rd, 10th, and 17th. To start off with, let me introduce our centre, the Advanced Centre for Chronic Diseases. Chronic diseases are leading causes of death worldwide and in Chile. As you can see from this graphic, the leading causes are problems due to heart disorders and circulatory problems and cancer. And this is no different in Chile. So here in Chile, you can see that again, cardiovascular diseases and cancer are the leading causes of death in the population. And so the objective of this centre, of our centre, is to provide a framework for the understanding and prevention of cardiovascular diseases and cancer, the two main chronic diseases affecting the Chilean population. And in association with a network of international and national collaborators, ACCDiS aspires to becoming a reference centre in Latin America for research and advanced training in chronic research, training in chronic diseases. So this is our history, a timeline started December 2013, renewed again for another 5 years, December 2018, and now we're here finishing year 7 in the centre. The centre is organised in the following way. We have 6 principal investigators: Sergio Lavandero, director of the centre, Pablo Castro, both work in cardiovascular diseases, myself and Alejandro Corbalan, working in the area of cancer, Caterina Ferrecchio, deputy director, working in cancer epidemiology, and Marcelo Kogan, working in the field of nanomedicine. Then we have a core project called the Mauli Cohort, or MAUCO. We have a data bank and a biobank there, and we have a common facility called the Basic Preclinical and Clinical Core Facility, which offers certain services to all participants in the centre. The centre is multidisciplinary, covers a wide variety of different areas, ranging from biotechnology, pharmacology, epidemiology, oncology, microbiology, nanomedicine, all the way to public health. The ACCDiS team now includes 246 people, a large majority of which are students in training, as you can see from this pie diagram. So there are undergraduate students, master students, PhD students, and a large number of postdoctoral fellows, in addition to, of course, professionals, collaborators, associate investigators, and principal investigators. And so in total, we have 60% of the people in the centre are basically people in training. Just briefly, some of our numbers. So to date, we have over 380 publications, average impact factor around 5.4, 220 or so Q1 journals, 70 or so top 10 in top 10 journals, and in addition, 14 patents. We also have a strong outreach program to society, involving, for instance, the use of Facebook, Twitter, websites. As you can see here, we have also newsletters in the centre, and we also have a new program called PAR Explorer, which was awarded to the center, and basically is a program dedicated to bringing science closer to children, school children, and the northern metropolitan region. As such, the center has organized over 300 different events with more than 30,000 participants. Research in the center focuses on the areas I mentioned, so predominantly digestive cancers in the area of cancer, and then heart failure in terms of cardiovascular diseases. Then we also have an area of nanomedicine and epidemiology, and several so-called transversal areas of research. So basically areas of research that connect these two rather distinct fields involving exosomes, angiotensin 1-9, gastric and gallbladder tumors, and also connections between cardiovascular disease and cancer. The strategy we use to promote such interactions involves the training of joint PhD, masters, and also students and postdoctoral fellows that work then on projects between 2 or more laboratories. And of course, we have the MAUCO cohort study, and we have specific internal funds that support such shared projects. The center has a large array of collaborations, both at the national level, so ranging from all the way from Arica in the north down to Valdivia in the south. And we further count on a large international network which spans essentially most continents in the US, Europe, Asia, Australia, and of course also collaborations in Latin America. So, and just to finish off here, centers in Chile were ranked recently by CIMAGO Institutions ranking, and they evaluated both centers like ours and also universities, and quite surprisingly, ACCDiS as a center with only 6 PIs, and of course the other 240 people, was ranked 3rd amongst research institutions, so all research institutions in Chile, just behind the 2 leading universities, the University of Chile and the Pontificia University, Catholic University of Chile. So with that, I would like to close this presentation of ACCDiS and move on to my own presentation, scientific presentation. Before doing so, just a couple of indications of how this is going to work. Each speaker in this session will give his or her talk. There will be no questions at the end of each session. I will just go on to introduce the next speaker, but there will be questions at the end of all the talks. So we have about 15 minutes there. So as we go through the talks, do write down your questions. You can also write them, the best is to write them down in the chat. And then at the end, as I said, in the discussion session, we will address those questions. We'll discuss them together with the speakers. Briefly to my person, as I said, I'm principal investigator in the Advanced Center for Chronic Diseases. By training, I was trained as a biochemist at the Swiss Federal Institute of Technology in Zurich. I got my PhD there, did 2 postdocs in the US, first at the University of Washington in Seattle, and then at Duke University in Durham, then moved on to, or back to Switzerland again as assistant professor at the Institute of Biochemistry in the University of Lausanne, and 5 years later moved then to Chile, so 1999, and formed the Laboratory of Cellular Communication, as you'll see in a minute, and I'm now full professor here at the University of Chile. So in my talk today, I will focus on discussing cell intrinsic and extrinsic mechanisms of covalent-1 enhanced metastasis. As I said, we are this laboratory, or form this laboratory called the Cell Communication Laboratory in the Faculty of Medicine here in Santiago in Chile. As such, we are part of the Center for Studies on Exercise, Metabolism and Cancer, so I'm actually director of that center, and also this center I introduced previously, the Advanced Center for Chronic Diseases. Our laboratory is actually 2 in 1. I share the space with my wife and her group, and she works on astrocyte neuron interactions, and you will hear more about that in the cell signaling session next week. So the 10th of November, the first session in the morning is another cell signaling session, which will be hosted by Lisette Layton. Or basically people in my group, work on a different area, obviously cancer, otherwise I wouldn't be here. Essentially, what we are interested in is the role of proteins or their function as tumor suppressant or oncogenes, and specifically how a protein called caveolin-1 can transit from one state to another. So the bottom line here of the first part is that caveolin is both a tumor suppressor and promotes metastasis. So as you can see here to the right, caveolin is a protein that inserts into the membrane with this hydrophobic loop, basically has a region, a carboxy terminus and amino terminuses that looks into the cytosol. And this is an important site, a modification of tyrosine residue 14 that can be phosphorylated, and we'll be hearing quite a bit about that in a moment. The protein dimerizes and then oligomerizes, associates with regions of the membrane called lipid rafts enriched in cholesterol and sphingolipids, and in doing so then forms invaginations of at the plasma membrane called caveoli. And so these are actually structures you can see in an electron microscope, as you can see here, an image of a fibroblast. So by definition, they are involved in things like vesicular transport and cholesterol homeostasis. The protein also binds cholesterol. But the role, of course, we're interested in is its role in signal transduction. And specifically what had been shown was that the protein was capable of interacting with a large number of targets that are signaling proteins that promote phenomena like, for instance, proliferation, survival of cells, which are obviously important characteristics in tumor cells. And the interaction with caveolin actually blocks this activity. So it was suggested quite some time ago that caveolin-1 was a tumor suppressor. This is actually how we got involved in the field. Just briefly here, one experiment in using HD29 human adenocarcinoma cells that lack caveolin. They form tumors in nude mice when injected subcutaneously. And if you introduce caveolin in these cells, the cells no longer form tumors, so clearly acting here as a tumor suppressor. What we did notice at the time, however, was that although caveolin is apparently initially downregulated during the process of tumor formation, it appeared that it could subsequently be upregulated again, for instance, in drug-resistant cells and also in metastatic cells. And so that led us to postulate that maybe it was doing different things depending on when it was expressed and who it was expressed with. And so this is essentially what we've been working on for the last 20 years or so. So this is just something we summarized in numerous reviews, where we show here in a schematic how caveolin initially in the transformation process is downregulated, and then later on, so in humans this could happen then 20 years later, can be upregulated again. And when it's upregulated and re-expressed, its presence coincides with things like multi-drug resistance and elevated metastatic potential. In summary, what we did to show how caveolin works as a tumor suppressor is found that it's present in a complex with E-cadherin, sequesters beta-catenin, and prevents thereby transcription dependent on beta-catenin TCF-LEF of important genes like cyclooxygenase 2 and survivin, and so in doing so reduces, for instance, cell survival and cell proliferation, and also things like angiogenesis. So this is the good side of caveolin. However, in cells where E-cadherin is absent, it actually develops a very different role, and this is what we're going to be mainly talking about today. So we're focusing on caveolin in these late stages, where, of course, over this period of time here, proteins like E-cadherin have been lost, so they've been they're basically being downregulated. So we're looking at cells now that do not have E-cadherin. And our entree into this part of the field came with this study where we showed that caveolin could actually enhance motility of cancer cells and focal adhesion turnover, that this required phosphorylation on tyrosine 14. To some extent, this had been shown by others, but what people had seen using different models, especially fibroblasts, is that caveolin presence and its role in migration was associated with accumulation to the rear of cells and, for instance, row activation. In our studies, we did not see such polarized accumulation, which led us to suspect that maybe caveolin would be doing something different in metastatic cells. Because in that study we saw that tyrosine 14 phosphorylation was important, we decided to generate mutants, so mutate this tyrosine either to phenylalanine, so a null mutant, or glutamic acid, so again a function mutant, and see what this or how this would change behavior. Just briefly, one in vitro experiment, which I think is kind of entertaining. So we obviously did the traditional transwell matrigel assays where caveolin promotes migration invasion, but it also does promote these traits in a matrix-specific fashion. And so this we studied using these so-called microfluidics chambers, where you grow the cells in the central chamber, and when they reach confluency, you actually then introduce liquid into these side arms, and this allows the cells then to migrate into these side arms, which were previously coated with specific matrix, extracellular matrix proteins like fibronectin, laminin, vitrogen, or collagen. And the kind of result you then get is what is shown in this little video here, where we can see that the caveolin expressing cells migrate more than the mock cells or the parental cells. And you can analyze different parameters of the migration of these cells. And you can see in doing so that caveolin promotes directionality and also increases velocity of migration of these cells. And it does so in a matrix-specific fashion. So you only see this when the cells migrating here on matrices, such as, for instance, vitronectin and laminin, but not collagen 4 or fibronectin. Now, let me just move on here to some in vivo studies, looking at this system where we use C57 black 6 mice and inject B16F10 cells, which is a highly metastatic cell line, into the either subcutaneously or into the tail vein, and we can look at both subcutaneous tumor formation and metastasis to the lung. These cells are characterized by the lack of caveolin and also the lack of E-cadherin. And so, as I said, when you inject cells now expressing or not caveolin, you look at subcutaneous tumor formation, or if you inject the cells intravenously, you can look at metastasis to the lung. And the result you get is summarized here. Looking at lung metastasis, you can clearly see how the presence of caveolin potentiates metastasis to the lung. And if we introduce this mutant, this Y14F mutant, this is no longer the case. Just as a control, we looked at subcutaneous tumor suppression, and cells expressing caveolin grow smaller tumors. And this ability is also maintained in the cells expressing the mutant caveolin. So the mutation doesn't affect the ability of caveolin to act as a subcutaneous tumor suppressor, but it does ablate the ability of the protein to promote metastasis. So this is the publication that emerged there from those studies, showing that indeed this behavior of caveolin-1 is matrix specific. So when thinking about migration, things that come to mind, there are small G proteins like Rho, CDC42, and RAC1. We wanted to know which one might be involved. In this assay here, we essentially let cells adhere to a surface and looked at Rho activation of different small G proteins and found that actually we specifically got increased activation of RAC1 and not necessarily of other small G proteins in those cells expressing caveolin. So these are breast cancer cells. And we get a similar result in these melanoma cells. So how do you connect now caveolin to RAC1? Insight came from a report into the literature suggesting that RAB5 can actually activate RAC1. And so we set out to test that using these colon carcinoma cells and melanoma cells in wound healing and transmigration assays. In black, you can see how caveolin in these different cell lines promotes or enhances migration in these two assays. But if we then generate a knockdown for RAB5, this ability is ablated, meaning that RAB5 is downstream of caveolin-1. And so this is the publication that emerged, showing that RAB5 is required for caveolin-enhanced RAC activation, migration, and invasion. And just to finish off this part, it is also required for caveolin-1 to promote metastasis. So again, here we have in black, you can see how the presence of caveolin promotes metastasis to the lung. And if we knock down in these cells RAB5, basically this ability is lost. So this is the schematic summarizing what I've mentioned so far. Caveolin phosphorylation connects to RAB5 via protein called P85, which is a gap. So by sequestering it, we enhance the activation of RAB5 of this GEF then TM1, which activates RAC1 and promotes migration, invasion, and metastasis. And now, interestingly, in this complex here with ACCDiS, caveolin is actually dephosphorylated. So the kinase that phosphorylates here is SARC. Here it's dephosphorylated, and this is the form of caveolin that contributes to its role as a tumor suppressor. And the reason it's dephosphorylated there is because a non-receptor tyrosine phosphatase is recruited to this complex that is called PTBN14 and dephosphorylates caveolin. This is something we just recently published. So caveolin-1 can either act as a tumor suppressor or promoter metastasis by cell intrinsic signaling mechanisms that vary in a cell context dependent manner. So just to finish off here briefly some of our work on caveolin in exosomes. And here we asked whether caveolin may be present in exosomes and what it might be doing there, whether it might be promoting migration and invasion of recipient cells. And the reason we came up with this idea is because it had been reported that in these MDA-MB231 cells, which we were actually working with, they produced exosomes and were able to convert MCF7 cells, which are a non-metastatic line, into a much more aggressive cell line by transferring exosomes. And since we knew these cells expressed caveolin and these didn't, we wondered whether caveolin might be involved in these processes. So exosomes or extracellular vesicles contain a large number of components, proteins, microRNAs, mRNAs. We'll focus just on the protein content and use some of the markers that are basically proteins involved in the biogenesis of exosomes as markers for our preparation. So CD9, CD81, TSG101, and ALIX, and as negative controls, ER proteins like calnexin and calreticulin. We use these MDA-MB231 cells that are short hairpin control, or short hairpin caveolin. So these cells express caveolin, these do not. We isolate the extracellular vesicles or exosomes using this protocol, and then characterize them either by nanotracking analysis, where you can see all three preparations are rather homogeneous in size, and also the quantities of vesicles produced are rather similar. And you can see that again here too. The different subpopulation size, subpopulations very similar, and the modes of are similar for the different preparations as well. Also, if you characterize them by electron microscopy, they look very similar. And indeed, if we then characterize these EVs by Western blotting, you can see that the cells that do not express caveolin also do not have caveolin in the EVs, the other ones do. These are just the marker proteins showing that our preparations do actually contain exosomes. And they do not contain markers of the endoplasmic reticulum. And so what we now did was to take these EV preparations and treat non-metastatic T47D cells and see how these treatments affected migration and invasion. And as you can see here, basically incubation with exosomes containing caveolin promote migration or invasion in these T47D cells, and actually also do so in MDA MB231 cells where caveolin was knocked down, so you get the same effect. Then to find out what might be causing this response of cells, we did mass spec analysis, characterized the 3 different preparations, classified these proteins here that are common to the vesicles with caveolin using gene ontology and Panther analysis, and we're particularly interested in those that then were found in the biological function called adhesion. Some of these are listed here. And by Western blotting, then we were able to show that indeed, confirm our mass spec analysis showing that SER61 and tenacin are not present in these preparations, vesicle preparations lacking caveolin, as you can see down here. Whereas for S100A9, there was still some protein in these preparations. So obviously, we're now focusing on understanding the role of these proteins. Finally, an in vivo experiment where we looked at the effect of these vesicle preparations on T47D cells, pre-treating them and then injecting the mice repetitively with these preparations, these vesicle preparations, and looking at retroperitoneal tumor mass and malignant acidic fluid formation. And as you can see from these results is when these cells are treated with vesicles containing caveolin, the tumor mass, the mesenteric tumor mass, is enhanced, not so if the cells are treated with vesicles lacking caveolin. And also the metastatic potential is enhanced as characterized by the number of cells present in malignant acidic fluid, as you can see here, is considerably enhanced in those cases where the vesicles contain caveolin. So let me conclude by saying that caveolin-1 expressing metastatic cells produce these exosomes containing caveolin. They convert benign tumor cells into highly invasive malignant cells, and they include specific cell adhesion molecules, which we think are important for this function. Of course, this is what we're studying now, and also how caveolin promotes the inclusion of these proteins into such vesicles. So in summary, caveolin promotes migration, invasion, and metastasis by cell intrinsic and extrinsic mechanisms. Just like to thank all the members of the lab here in a retreat in our house up in the Cajon de Maipo. Those were the happy days back in 2019, or before we had all the social unrest in the country, followed then by the pandemic in 2020. And so now our lab meetings and gatherings look more like this, very similar to our gathering here today. Thank our collaborators, national, international collaborators, and of course funding, and you for your attention. As I said, we will be taking questions at the end of all the talks, and so I will move on now to introduce our next speaker. Do remember to add your questions to the chat, and we will discuss them at the end. Okay. So our next speaker is Vicente Torres. Vicente Torres got his PhD in biochemistry from the University of Chile in 2006. He then did his postdoctoral training in the Cancer Center of UC San Diego, 2006 to 2009, with Dwayne Stupak, looking at the role of caspases and endocytic trafficking in tumor cell migration. He then moved back to Chile to the Faculty of Medicine in 2010 as an assistant professor, and moved on then to the Faculty of Dentistry, again in the University of Chile, where he's now associate professor and director of the PhD program there. And he's going to tell us today about early endosome cell migration and the epithelial to mesenchymal transition, new connections, and their contribution to tumor progression. Vicente, go ahead.
Thank you, Andrew, for this presentation, and good morning to everyone. And I thank for the invitation to give this talk in this meeting. And what I'm going to talk to you is about the early endosomes, cell migration, and the epithelial to mesenchymal transition, and what are the new connections and their contribution to tumor progression. Cell migration is a fundamental process in a multicellular organism, as it is involved in the embryonic development, in the generation of immune responses, during the wound healing process, for instance, when we have the re-epithelialization of epithelia, and in pathology where it's deregulated in cancer and metastasis. Okay. So I'm going to give a brief introduction to the tumor microenvironment, specifically the hypoxic tumor microenvironment. And this is characterized by the low oxygen levels, the decreased supply of nutrients, and poor vascularization. This tumor microenvironment is an adaptive force for pushing cells to invade other tissues. So hypoxia has been associated with increased malignancy, poor prognosis, and metastasis. How hypoxia might be connected with in promoting these events leading to metastasis? Mechanisms are not clear completely yet, but it is known that it involves in promotion of cell migration and invasion, changes in the availability of cell surface receptors, intracellular signaling, and more recent works have linked hypoxia with changes in the intracellular trafficking. I'm going to pay attention to this very last point because deregulated intracellular trafficking has been considered an emerging feature of cancer, because it is known that endocytosis of cell surface molecules such as ACCDiS is relevant for the epithelial to mesenchymal transition. Also, it is known that signaling endosomes, which are in this part of the scheme, are important in signaling from the inside of the cell when we have, for instance, growth factors. And interestingly, for us, it has been shown that intracellular trafficking is associated with the cell attachment, detachment, and also for cell migration. Specifically, you have here a cell adhesion molecule that is going to be internalized and then recycled back to the leading edge of a migrating cell. So we can expect that this system is deregulated in tumor cells. In this video, you can see the migrating cell forming new cell adhesions and disassembling pre-existing cell adhesions labeled in green here, according to the movement of the cell. And what we observed in back in 10 years ago, these are former studies from the lab, that when you have a cell migrating, this is a lung carcinoma cell in culture, you have this polarization, the leading lamella, And if you pay attention to the early endosomes, which are decorated for this protein called RAP5, you have these early endosomes accumulated at the leading edge. So what are these early endosomes doing in this part of the cell? And why is this relevant in the context of cell migration? To this end, I'm going to talk to you about the RAP proteins. RAP proteins are more than 60 members in humans, and they are involved in the control of trafficking, endosomal trafficking. For instance, we have RAP5 that controls early endosome trafficking, RAP11 and RAP4 in the recycling pathways, and RAP7 in the late endosome degradation pathway. As small GTPases, the RAP proteins cycle between two states: an inactive state that is GDP bound and active state that is GTP bound. This cycle is promoted by GEF proteins, which activate the RAP proteins, and by GAP proteins, which inactivate the RAP proteins. So the first question that we were wondering is on whether this, because we observed accumulated early endosomes at the migrating cells, we were focusing on this protein, RAP5, which could be activated during cell migration. In this case, we perform a pull-down assay in migrating cells in a multi-wound scratch assay. As you can see here, this is a pull-down assay. We have an increased activation of RAP5 during cell migration. Now, this is a very summarized model that we work on those initial years that propose that RAP5 in the early endosomes promote the disassembly of focal adhesions and the new formation of focal of new adhesions at the leading edge of migrating tumor cells. I'm going to show just a few experiments supporting this model, just to have clear the picture. And here, this is back in 2010, we observed that RAP5 is accumulating at the leading lamella and colocalizes with beta-1 integrins at the leading edge of the migrating cell. What is doing in this region? We have early endosomes labeled for RAP5 in green here at the protrusions, at the cellular protrusions, but also at the detaching sites of the cell when it's stimulated to migrate. And specifically, these endosomes, which are observed in green here, colocalize with focal adhesions, which are the structures that permit attachment to the extracellular matrix. And you can see here a very restricted colocalization between early endosomes and the focal adhesion. Importantly, if you see in this video, this focal adhesion, when it's colocalizing with the early endosome, is followed by their disassembly, and this is relevant for cell migration. It is known that focal adhesions, which are the structures that I showed in here, that are formed by a group of proteins, are disassembled by proteolysis. And it's been shown that calpains are involved in the proteolysis of this protein at the focal adhesion complex. What we observed in more recently is that early endosomes, which are labeled here in red with the EEA1 antigen, are decorated with calpain 2 at the periphery of the endosome. This is a 3D reconstruction. For reasons of time, I don't show you much data about this, but we were able to observe that when you knock down either RAP5 early endosome recruitment of calpain 2, we prevent cleavage of these proteins, and cells are no longer able to migrate. Now, coming back to the hypoxic tumor microenvironment, as you remember, hypoxia promotes cell migration and invasion. So the question that we were wondering is on whether hypoxia could be able to stimulate this mechanism of regulation of cell migration and focal adhesions. So, is hypoxia connected with the early with the endosome dynamics and cell migration? To this end, we picture that okay, a cell migrating, tumor cell migrating, when it's exposed to hypoxia, we expect to observe increased cell migration. That is suggested in the literature. In fact, we observe that when lung carcinoma cells are exposed to hypoxia, RAP5 is increased in its activity. And this is associated with an increase in cell migration and cell invasion. Importantly, when we knock down RAP5 in these cells, we don't observe any more hypoxia-induced cell migration and invasion. If you remember this model, focal adhesion disassembly, interestingly, when cells are exposed to hypoxia, there is an increase in the colocalization between RAP5 and the focal adhesion protein paxillin that you can see in here. This is in hypoxic conditions. And this is corroborated in vitro by biochemical, by immunoprecipitation. When you see here RAP5 co-immunoprecipitates with vinculin and tallin, and this co-immunoprecipitation is increased in hypoxic conditions. The next question that we wonder is whether RAP5 could be associated as an indicator of the aggressiveness of a tumor cell to invade and metastasize. So we use this model, this isogenic model of mouse melanoma cells that are highly metastatic and poorly metastatic. And we expose these cells to hypoxia and measure RAP5 GTP levels. And as you can see here, RAP5 got activated only in the highly metastatic cells following hypoxia. And this was associated with a significant increase in cell migration in this model. However, if we overexpress the wild type form of RAP5 in this poorly metastatic cell line, we are able to sensitize these cells to hypoxia-induced migration. Conversely, if highly metastatic cells are transfected with a inactive mutant of RAP5, there is an impairment in hypoxia-induced cell migration. Then we use the same model as a tail vein injection in mice for measuring lung metastasis formation. And as you can see here, hypoxia increased lung metastasis in these mice made with the B16F10 cells. And when we knock down RAP5 in these cells, we were not able to see any more hypoxia-induced metastasis in this model. Therefore, the model that we were working on is, this is a very summarized model, and there is no data that I didn't show all the data in here, but basically RAP5 promotes focal adhesion disassembly, then in stimulating cell migration and promoting metastasis. And hypoxia increases this activation of RAP5. So the next question was on whether how hypoxia activates RAP5. To this end, we have this transcription factor, HIF1 alpha. HIF1 alpha is stabilized, is a transcription factor that stabilizes in hypoxic conditions and leads to the transcription of target genes. So in our models, HIF1 alpha was stabilized in hypoxia, and this was prevented in cells treated with an siRNA targeting HIF1 alpha. Interestingly, RAP5 got activated in hypoxic conditions, but this was prevented when we knocked down HIF1 alpha. In agreement with those findings, this whole expression of HIF1 alpha, this is a mutant that is stabilized HIF1 alpha, was able to increase RAP5 GTP levels. And this was also observed in a model of endogenous stabilization of HIF1 alpha by mutation of the VHL protein. you can see here an increase in the activation of RAP5. Okay, so HIF1 alpha is a transcription factor and is required for RAP5 activation. So the next question was on whether this was a transcriptional mechanism. To this end, we use an pharmacological inhibitor of HIF1 alpha, and as you can see here, the activation of RAP5 in hypoxia was prevented by this inhibitor of HIF1 alpha. This suggests that the activation of RAP5 depends on transcription. Interestingly, total RAP5 did not change in any condition. Hence, we hypothesize that any intermediate factor such as a GEF or a GAP could be induced or repressed in hypoxia that accounts for RAP5 activation. And this is a selected data showing that a subset of GEFs were induced in hypoxia. These include RIN2 and alsin2. These are GEFs for RAP5. Interestingly, the GEF alsin2 contains an HRE, which is an hypoxia response element that are putative, and one of those are functionally active in binding HIF1 alpha in hypoxia. Then we checked on whether hypoxia indeed increased the protein alsin2. And as you can see here, by Western blotting, alsin2 was increased in hypoxia. And this was also observed in the endogenous model of stabilization of HIF1 alpha. Most importantly, alsin2 induction was required for tumor cell invasion and metastasis, because when we knock down alsin2, tumor cell invasion is decreased, and this is also observed at the metastasis experimental model, whereby hypoxia increased tumor cell metastasis, but the knockdown of alsin prevented this increase. And this is a quantification of this data. Finally, we checked the expression of these proteins in tumor samples in patient biopsies. And this is a clear cell renal cell carcinoma samples that you can see increase in HIF1 alpha expression, as previously known, but this is new, and we found that alsin2 expression is associated with an increase in HIF1 alpha expression in tumor samples. In fact, when you see at the very in detail in a tumor, you will see that alsin2 is increased only in the tumor region, but not in the non-tumor region, as shown from two patients in here. So with this, I'd like to finish this model that RAP5 promotes cell migration and focal adhesion disassembly. This is stimulated by hypoxia, which stabilizes HIF1 alpha. HIF1 alpha induces alsin2, which is responsible of the activation of RAP5. Just to finish up, I'd like to take a couple minutes, the last 2 or 3 couple minutes that I have, to tell you about that the deregulation of RAP5 activity is not only observed in cancer cells. More recent studies from our group have shown that RAP5 is also activated in potentially malignant disorders. To this end, I'm going to show you very quickly this model, oral carcinogenesis in the oral mucosa. Here you have a stage that is called dysplasia, and dysplasia is considered as a highly predictor of transformation to carcinoma. And what this is a summary of the findings that we have showing that RAP5 gets activated during the progression of oral dysplasia, and this is associated with the increase in early endosome size. There's also other markers that I'm not going to discuss at this point. But here you have a data showing that the early endosomes, which are stained in green, this is a tumor sample, a biopsy by immunofluorescence, showing the size of the early endosomes, which are progressively increased in oral dysplasia. When we moved to an in vitro model, which is oral dysplastic keratinocytes immortalized, we observed an increased activation of RAP5. And when you express a mutant of RAP5 in this model, there is a decrease in genes typically associated with oral dysplasia, which are survivin and cyclin D1. Interestingly, these two genes are dependent on a common pathway, which is the beta-catenin signaling pathway. And the beta-catenin signaling pathway involves the stabilization of this transcription factor, beta-catenin, which translocates to the nucleus and drives the expression of these genes. We know previous studies from our group show that beta-catenin accumulates at the nucleus of oral dysplasia. This is in patient sample. And interestingly, in the in vitro model, when we express a mutant of RAP5, this mutant, here, you will observe a decrease in nuclear beta-catenin in transfected cells. And to conclude about the mechanism, and this is the very last slide, we propose this model whereby in dysplasia, there is a stabilization of beta-catenin, which is associated with the sequestration of the destruction complex of beta-catenin within early endosomes, and then multivesicular bodies. This is only a panel showing the increase in the early endosomes in green, and these early endosomes colocalize with the destruction complex, and this colocalization is increased during oral dysplasia, which is summarized in this model. As you can see here, an increased sequestration of this complex in oral dysplasia. With this, I like to bring you the take-home message that the deregulation of this system is not only in cancer cells, but also in early lesions. Thank you very much. I'd like to thank the ACCDiS for this support and my laboratory and the collaborators of this project. Thank you very much.
Okay, thank you very much, Vicente, for that presentation. As I mentioned, there will be no questions at this point. We will take questions in a roundtable discussion at the end. Please jot down any queries you might have in the chat, and then we'll sort of address them, as I said, later on. So with this, we will move on to our next speaker, who is Fabiola Sanchez. Fabiola Sanchez got her PhD in, Vicente, can you sort of cut the video? Thank you. So Fabiola Sanchez got her PhD in 2004 in cell and molecular biology at the Catholic University of Chile. She then went on to do a postdoc with Walter Duran at the New Jersey Medical School, studying the role of endothelial nitric oxide synthase localization in endothelial permeability. She then moved back to Chile, took up a position in the Universidad Austral de Chile, so down in Valdivia, and she now holds a position there as associate professor. And she will now talk to us about signaling mechanisms regulated by S-nitrosylation in the endothelium and its contribution to extravasation and metastasis. Welcome to the session, Fabiola.
No se escucha, Fabiola. Está sin...
So sin micrófono, Fabiola.
Sorry about that. I will start again.
Okay.
So, thank you very much for the invitation, and I will start my presentation today talking about nitric oxide, which is a gas molecule that regulates many functions in the organism, as you can see here. And I am particularly interested in the regulation of endothelial function by nitric oxide. Well, after stimulation with different types of stimulus, these nitric oxide synthase that are expressed in different cell types induce the nitric oxide production, and this nitric oxide can activate two pathways. One is the 1-allyl cyclase and PKG pathway, which is called the classical pathway, and the other one is the S-nitrosylation, which is the binding of a nitric oxide group to free thiol in cysteines to form an S-nitrosothiol that we and other groups have demonstrated that regulates the traffic of protein, interactions, and phosphorylation of proteins. Generally, to abort this problem, you have to do site-directed mutagenesis of the proteins that are S-nitrosylated to see the real effect of the modification. But you can also abort this problem at the beginning by using two inhibitors. One of them is LNMA that inhibits the nitric oxide production, and the other one is ODQ, which inhibits the classical pathway. If one effect is inhibited by LNMA, but not by ODQ, it strongly suggests that the active pathway is the S-nitrosylation. And we use this approach because up to now, there is no specific inhibitor of this pathway. Alternatively, we have used N-acetylcysteine, NAC, which is an antioxidant that indirectly inhibits S-nitrosylation. And we have used it because NAC is a FDA drug, approved drug used for the treatment of overdoses with acetaminophen and in lung obstructive disease. So it can be easily repurposed for other diseases. So as I said before, I'm particularly interested in endothelial regulation, and as you can see here in this picture, endothelium play an important role in the metastasis and allowing both the intravasation of tumoral cell, and I will focus in this process which is the extravasation. And you can see that in order to the tumoral cell to get through the endothelium and form the metastasis, they have to interrupt the endothelial bindings. And by studying inflammation, we published a series of papers, and we came up with this model of dissociation of the endothelial barrier in response to pro-inflammatory cytokines, in which after stimulation with different type of stimulus pro-inflammatory, enos moves from the caveoli to the cytosol and then S-nitrosylate this adherens junction protein that keeps the endothelial barrier, the endothelial cells together. These proteins are S-nitrosylated, and because of that, some of them get phosphorylated, then they dissociate each other, and finally they get internalized. And in this way, there is an increase in permeability. We have identified the site of S-nitrosylation in P120. Another group has identified the sites in beta-catenin. Well, we thought that this process can also be mediating the pass of tumoral cells in the endothelium. And this is because there is a strong correlation among permeability and extravasation of tumor cells and metastasis. But before the cells gets into the endothelium, past the endothelium, they have to bind first to the endothelium. And this paper came up in 2016, and that it showed that in isolated vessels, tumoral cells that are labeled in red are bound to these sites in the microvasculature. These are curved zones in the microvasculature. And this correlates with the level of nitric oxide labeled with this proof. And you can see that these zones that are very rich in nitric oxide are the zones where the tumor cells are bound. When they inhibit the nitric oxide production using LNMA, they lower the nitric oxide levels, and this correlates with less addition of tumoral cells. So it looks like nitric oxide is also mediating the addition of tumor cells to the endothelium. And well, tumor cells adhere to the endothelium using these addition proteins like S-selectin, BCAM1, ECAM1, that are the same proteins that leukocytes use during the leukocyte activation cascade that causes transmigration of leukocytes. So even though these processes are not the same, they share some regulatory components. So we start to look for leukocyte addition in a way to, after we investigate here, that is a model more known to us, translate the discoveries in this process to tumor cell addition. So I will show you now some experiments that we have done in leukocyte addition. So we use this model to study leukocyte addition. This is the cremaster model that is very beautiful and used to us because it allows the observation of processes in real time. And you can see here that the cremaster muscle is a very irrigated tissue and is also very transparent, so you can easily put it under the microscope. You can get movies like this is a very short movie. This is a blood vessel, and we apply a pro-inflammatory cytokine on it, and you are going to see the binding of tumor cells to the endothelium. Here you have one cell that is binding. There are some that are just passing through, but in the time, more and more cells are bound to the endothelium, and this process occurs very fast also. So you have many leukocytes bound there. Well, you can quantify these images, and when we did that, we found that both cytokines, interleukin A and TNF alpha, they increase leukocyte addition in a very significant way, and this increase was inhibited in the presence of LNMA. And this increase was very fast also. And we use these cytokines because they are highly produced by tumoral cells. We also did it in vitro by incubating endothelial cells with leukocyte isolated from healthy donors. And again, we found that both cytokines increase leukocyte addition in a significant way. And this increase is inhibited by LNMA, but not by ODQ. And if you remember from my diapo at the beginning, this fact strongly suggests that the process go through S-nitrosylation. So how S-nitrosylation can be regulated leukocyte addition? Well, we focus in the protein addition expression, but we discard transcriptional regulation because of the timing of the effect that is very fast. So we studied two possible mechanisms that regulate protein addition expression in endothelial cells. And one of them is clustering, by which addition proteins in the cell surface are regrouped and they interact with the cytoskeleton, and this process doesn't change the total amount of addition protein in the cell surface. It just changes the affinity of the addition protein for the integrin in the leukocyte or tumoral cells. And we also studied traffic of proteins in which addition proteins in vesicles are transported to the cell surface. And in this mechanism, the amount of protein in endothelial cell surface is increased. So we start to looking for ICAM1, what happened with ICAM1. And this is a subcellular fractionation experiment. And here we found that the amount of ICAM1 in the plasma membrane, it does not change after stimulation with TNF-alpha. So it should be clustering. So to corroborate that, we watch for the interaction between ICAM1 and cortactin, a cytoskeletal protein, and we observe that the interaction is increased in the presence of TNF-alpha, but is inhibited in the presence of the NOS inhibitor. We also corroborate that in a more elegant way by doing FRAP, fluorescent recovery after photobleaching. And in that experiment, we transfect endothelial cells with ICAM-GFP. Then you do a small bleach with the laser in the microscope, and you measure the time that that bleach is recovered. And the whole reasoning here is that when the protein do clustering, this association with the cytoskeleton gets the protein unmobile in the cell surface, diminish the mobility of the protein. And you can see that with TNF-alpha, after treatment, the bleach is done and that bleach takes longer to recover. I'm going to show you here the curves of recovery. And you can see here that this is the recovery of fluorescent for control cells. But the cells that have been treated with TNF-alpha, they don't recover the fluorescent. But if you do the experiment in the presence of LNMA, the inhibitor of NOS, the recovery is, again, like control. So it means that this process of clustering is being regulated by nitric oxide. So we went to investigate if ICAM1 was nitrosylated, and we found that it is not. Okay, so this is also a message for all the people, that many people think that all the proteins are S-nitrosylated, and is not true. Well, this brings us a problem because we start to look for other possible proteins that can be S-nitrosylated and be regulating this ICAM1 clustering. And as you can imagine, there is a lot of protein that can be regulated by S-nitrosylation. But we focus our attention in protein kinase C theta, because this protein is involved in ICAM1 clustering. In response to TNF-alpha, PKC theta moves to the plasma membrane and phosphorylate ICAM1, inducing its clustering. And PKC theta has many cysteine in their sequence, and also had been demonstrated to be its activity to be modulated by N-ethylmalamide, which is a blocker of thiol groups, and thiol groups are where the S-nitrosylation occurs. So for that reason, we focus our attention in PKC theta. And we found that the S-nitrosylation of PKC theta is increased in response to TNF-alpha, and this is inhibited by LNMA. We also measure the PKC theta activity as is phosphorylated form that is increased, and this effect is also inhibited by LNMA. We also check the interaction between PKC theta and ICAM1 by co-immunoprecipitation, and this interaction is increased in the presence of TNF-alpha, but is inhibited in the presence of the NOS inhibitor. And finally, we check for the phosphorylation of ICAM1, that is phosphorylated in the presence of TNF-alpha, and this phosphorylation is inhibited in the presence of LNMA. Well, now we want to see which are the site of S-nitrosylation of PKC-zeta. And at the beginning, we were not very optimistic because PKC-zeta has about 13 systems. So imagine if all of them are S-nitrosylated, we have to do many site-directed mutagenesis. But we were lucky to find that only one system is S-nitrosylating PKC-zeta. This was done by incubating purified PKC-zeta with nitric oxide donors and then doing mass spectrometry. And this system here is located in this gray region that is in the kinase domain of PKC-zeta. So it makes sense that the S-nitrosylation can be regulating then the activity of PKC-zeta. And we have now the mutant of this site, and we are doing the experiment to test the loss of function of this mutant in leukocyte adhesion and then in tumor cell adhesion. So the other protein that we investigate was BCAM1, and we use interleukin A, other pro-inflammatory cytokine highly expressed by tumoral cells. And we found that in response to interleukin A, the amount of BCAM1 in the plasma membrane, it does change. So this can be modulated by traffic, in contrast to ICAM1. This is the inhibition in the presence of LNMA. You can see that the increase in the plasma membrane is no longer seen in the presence of the NOS inhibitor, and also there is an increase in the S-nitrosylation of BCAM1 in response to interleukin A, in contrast to what happened with ICAM1 that was not S-nitrosylated. So we went back to tumoral cells, and using now in our experiment conditioned media from tumoral cells, because our supposition is that the same tumoral cell is secreting this agent that are acting on the endothelium. So we took a conditioning media from MDA MB231 cells, and we measured the leukocyte adhesion in vivo, and we found that the media strongly induced leukocyte adhesion that is inhibited in the presence of LNMA. The conditioning media also induced the expression of BCAM1 in the cell surface that is inhibited by LNMA, and conditioning media also induced S-nitrosylation of BCAM1 which is also inhibited in the presence of LNMA. And then we want to see what happened now with the transmigration of tumoral cells through an endothelial monolayer in vitro. So we incubate MDA cells labeled with M-cherry. We incubated with endothelial cells, and after a while we took the filter here and by immunofluorescence see the tumoral cell that have passed the endothelium. And you can see here the cells in red, the nucleus are in blue. There are many cells that have passed through the endothelium in control, but when the endothelium is treated with NAC or with LNMA, two inhibitors of S-nitrosylation, there is a very high diminished transmigration. And we also moved this result to an in vivo model to see what happened in an in vivo model. And we use this model of breast cancer, a very aggressive breast cancer, the 41 model. In that model, the primary tumor is formed in 3 to 4 days. And after the primary tumor is formed, we start to give different treatments. We treated with NAC and with LNMA. And after the day 28, we examine the metastasis. Here you can see the formation of the primary tumor. And we observe the superficial metastasis, which are these small like vesicles in the lung from one side of the lung and the other, the opposite side, and we count them. And we found that the treatment with NAC decreases significantly the amount of metastases. And for LNMA, there are a tendency to slow the metastasis, but it's not significant yet. We also did an immunohistochemistry of the lung to see S-nitrosylated proteins, and this is the label for control lung. You can see that the lung with tumor has a very big increase in the amount of S-nitrosylated proteins, and the animals that were treated with NAC, they have a similar or less label than tumor cell, very similar to control. And also the histologist said to me that this is very similar as a healthy lung. And we have also measured the S-nitrosylation of protein in this lung lysate, and we've found so far that BECAM gets strongly S-nitrosylated, and LNMA and NAC, they both have decreased the level of S-nitrosylation of BCAM1. So here we have to measure also the level of nitrosylation of PKC-zeta and also of the adherens ganglion protein. So with all this data, we have established, or we propose, not established, we propose this model in which after the primary tumor is formed and the tumoral cell gets into the circulation, they secrete many cytokine or agents that can activate receptors on the endothelium, and this can activate nitric oxide production that may induce the traffic of endothelial adhesion proteins to the cell surface, or can also S-nitrosylate PKC-zeta to induce the clustering of protein in the cell surface, and these both processes can then bind tumoral cells. And then for the S-nitrosylation of the adherens ganglion here, this tumoral cell can go through the endothelium to form the metastasis. So that is my presentation, and I would like also to thank all the people in my lab that have contributed to do this work, and collaborators, international and national, and also funding. Thank you very much.
Okay, thank you very much, Fabiola, for your presentation. Again, as said at the onset, please jot down your questions in the chat and we will discuss them at the end of the session in the roundtable discussion period. And so we will now move on to the next presentation by Dr. Roxana Pincheira. Roxana got her PhD in 2000 in biomedical sciences at the University of Chile. She then went on to do her postdoctoral training with David Donner at Indiana University, Purdue University, in the cell signaling surgery oncology lab, and apparently then moved on for another stage at the University of California, San Francisco, looking at processes of cell signaling in cancer. Then she moved back to the Universidad de Concepción, where she is now associate professor. And she will talk to us about CEL2, a double agent transcription factor in cancer. Roxana?
Okay, good morning to everyone. Greetings from Concepción. Here in the map you see we are in the Biobío region. I would like to thank especially to Dr. Andrew Quest for this invitation to such an important event for the ACCDiS group, especially in the Cancer Symposium 2020. I would like to keep the video off, if it's okay, Andrew? Okay, it's because there is a message here.
Yes, yes, by all means, you can turn off the video to improve stability of the transmission.
Yes, but I don't know why it say iniciar video there. There is a message. Can you turn off that?
Where is that message? In the chat?
No, in the screen, but that's okay. I don't know why it's that. Okay.
No, just turn off your video there if you don't want to use it.
Okay. Okay. So like other researchers, we are interested in the understanding of the basic mechanisms of cancer biology, but the main focus of our lab is in transcription factors. Transcription factors, as you know, are proteins that play important roles in gene regulation. They are usually present in the nucleus. They bind a specific DNA sequence in the genome, but many transcription factors are also present in the cytoplasm as a latent transcription factor, so they need signals to become activated and move into the nucleus. They are involved in all the different cellular processes, such as development, proliferation, cell survival, cell differentiation. They are important for tissue-specific regulation, as well as in response to stimuli. Sorry, there is something wrong. The problem becomes when transcription factors are altered. This happens in cancer, so there is different things that can happen. They can be deregulated by different mechanisms. That means they can be upregulated or downregulated in relation to the normal tissue. They can be mutated, which can cause hyperactivation or inhibition of the transcription factor. They can be silenced or deleted, which is going to mean there is going to be loss of function of these transcription factors, or they can be fused with other genes. All these things can impact in cancer biology and the hallmarks of cancer. An example of transcription factor in cancer belong to the two main groups of genes related to proto-oncogenes and tumor suppressors. So in the proto-oncogene, we have MYC, which is usually found hyperactivated and amplifying cancer, while in the other side of the balance, we have the tumor suppressor p53. Both proteins are altered in more than 50% of human cancers. So in addition to the group of the oncogenes and tumor suppressor genes, a recent article studying genetic components in cancer development through database search and text mining identified a third group of genes, the POTSF, standing for proto-oncogene tumor suppressor factors. In this group, interestingly, more than 50% of the genes encode for transcription factors. You can compare that in oncogenes corresponding transcription factor 12% and in tumor suppressor gene 14%. So in this group, most of them are transcription factors, and they play important roles in gene regulation, acting as a negative and positive regulators. They are mostly present in the nucleus, and as expected, they have this dual role as proto-oncogenes and tumor suppressor genes. We are studying a poorly characterized transition factor that we believe belong to this group of POTSFs. So the factor that we are studying is SAL2. It is a member of the SAL-SPAL family of transition factors that are characterized by the presence of a conserved repressor domain in the N-terminal, a C2HC zinc finger motif that has not been characterized, a glutamine-rich region important for protein-protein interaction, and several C2H2 zinc finger domain along the protein. There is a large protein between 900 to 1500 amino acids and are conserved. They are also present in Drosophila and C. elegans. And in vertebrates, there is 4 members, SAL1, 2, 3, and 4. And SAL2 is the most disseminated member of the family, especially in the C-terminal domain. SAL proteins play key roles during organ development, and therefore they have been associated with genetic disorders. Just to give you some examples, SAL1 is associated with Townes-Brocks syndrome, and SAL4 is associated with Haukihiro syndrome, both of them related to heterozygous mutations in these genes. SAL2 gene was characterized by Ma in 2001. It is located in the chromosome 14 in a region where there is loss of heterozygosity in several cancers. It contains three exons, exon 1, exon 1A for alternative exon 1, and exon 2. And it has two promoters, P1 and P2. The gene can encode, depending on the promoter, two different alternative isoforms that are very similar, and the only difference is at the N-terminal domain, where isoform E1 is the one that contains the concept repressor domain, and isoform E1A, it doesn't. E1 is tissue-specific, and E1A is ubiquitously expressed. In relation to functional domains in SAL2 protein, 3 domains has been identified. In the C-terminal domain, several years ago, it was a paper that identified domain on SAL2 that interact with the large T antigen from polyoma virus and relates SAL2 to a tumor suppressor function. The same domain is important for transactivation activities. The DNA binding domain is in the central part of the protein, corresponding to this 3-motif C2H2 zinc finger domain that, according to a select procedure, supposed to bind to GC-rich regions similar to SP1 transition factor. And several years ago, we identified a domain in the N-terminal domain that is important for the interaction of SAL2 with the P75 neurotrophin receptor that is also belong to the TNF receptor family, and it relates to a functional, neural function of SAL2. So evidence indicates that SAL2 plays a role in neural and eye development and also in neuronal differentiation. The SAL2 knockout mice will show background-specific neural tube defect, as you see here. There is a problem in the closure of the neural tube, suggesting that SAL2 has a role in neurogenesis. We show that in the context of the interaction of SAL2 with the P75 neurotrophic receptor, SAL2 is important for neuronal differentiation. You can see here in this picture that hippocampal cells that doesn't have SAL2 cannot extend neurites, so there is no projections and there is no dendrites or axons, so the function of these neurons is abolished. Recently, this year, a new paper came out indicating that the same signaling we identified several years ago is involved in the maintenance, the stemness of conjunctival epithelial stem cells and in regulating differentiation status. And Kevelman in 2014 identified SAL2 homozygous mutation as the cause of the recessive ocular coloboma, that is an eye disease that causes blindness. And the mutation specifically is in the codon 85 that generate a stop codon. So there is not, it's a truncated protein and the DNA binding domain is not present in this truncated protein. But why I'm here today is because SAL2 is altered in cancer. So transcriptomic, macroarrays, and tissue array studies have demonstrated that SAL2 is downregulated in a number of cancer, while it's also upregulated in a different cancer types, such as synovial sarcoma, with tumor testicular cancer, oral cancer, and glioblastoma, just to mention some of the data that exists. In addition to the deregulation of SAL2 levels in cancer, from CEBE bioportal, it is found that from 23 cancer types, SAL2 is altered in 24 of them by different genetic alterations. Interestingly, in some cancers, SAL2 is found just deletions of SAL2. In other type of cancer, there is mainly amplifications, and while in others, we see mainly mutations. In other cancer types, you see many different kind of alterations in SAL2 genes. So the significance of that is unknown, but definitely indicate that SAL2 has something to do with different cancer types. As the title of my talk says, SAL2 as a double agent, that means these two are all similar to caveolin, I guess. And I'm going to show you some examples of the role of SAL2 as a tumor suppressor in the context of ovarian cancer and in normal cells, while there is also evidence that SAL2 plays a role as an oncogene in the context of glioblastoma. So in the context of glioblastoma, there was this seminal paper from Suba published in Cell in 2014, trying to identify what factors were essential for the reprogramming of differentiated glioblastoma cells into a stem-like tumor-propagating cell that is the aggressive phenotype cells. In this context, this group identified 4 factors, OLIC2, PUF, SOX2, and SALT2, as the key factors for the reprogramming. So this was the main finding, and additional papers after that have demonstrated that SALT2 is definitely amplified in the context of glioma. And they are trying to identify what is called the switch genes that can turn off this program. So by bioinformatics and computational analysis, they identified that phospholigand-1, a transcription factor of the phos family, is the switch gene that can turn off the expression of the 4 neurodevelopmental transcription factors, including SALT2. So currently, phospholigand-1 is a potential target for glioblastoma because of the importance in turning off the neurodevelopmental transition factors. In the context of ovarian cancer, SALT2 is considered as a tumor suppressor. There is loss of heterozygosity in a number of ovarian cancers. And in 2004, Lee and collaborators found that ovarian cancer cells express low levels or no expression of SALT2 compared to the normal epithelial ovarian cells. And when these ovarian cells express SALT2 and are injected into seed mice, they realized that the tumor size and volume decrease, and this was caused because a decrease in the percentage of mitotic cells, and correlate also with an increase of the apoptotic cells, indicating that SALT2 may be playing a role as a tumor suppressor in ovarian cancer. Additional studies in human samples have demonstrated that in effect, SALT2 is present in epithelial normal ovarian, but not in the carcinoma. And there is this study of 2010 cases of ovarian carcinoma. In different histological subtypes, you see that SALT2 expression is negative mainly. Why is that? They also found that one important factor for SALT2 downregulation relates to hypermethylation of the promoter 2. So the tumor, but not the normal tissue, is hypermethylated, and this because the P2 promoter contains several CpG-rich regions. So this is one of the mechanisms for loss of SALT2 expression in ovarian cancer. We have been working using a SALT2 knockout model by collaboration with Dr. Ruichi Nichikamura from Japan. So this model, of course, we don't have expression of SALT2. We have been doing mainly in vitro studies to figure it out SALT2 function under normal conditions. And we have been interested in understanding how SALT2 is regulated, as is deregulated in cancer, so we need to know how it's regulated in the normal context, what targets and cellular functions. But today I'm just going to show a little bit of our findings in the context of targets and cellular function of SALT2. Several years ago, we published the SALT2-induced apoptosis under genotoxic stress. Here you see this is in vitro and this is in vivo. We see that under genotoxic stress induced by doxorubicin, that's a chemotherapeutic agent, wild-type cells, apoptosis is induced. But we noticed that in the context of the SALT2 deficient cells, apoptosis will dramatically decrease. In the context of radiation with gamma radiation of the mice, and then we collected samples and analyzed radiosensitive tissues. So here we see the example of the small intestine. SALT2 wild-type mice, you see have an increased apoptosis detected by the tunnel assay, but this was not happening or was decreased in the context of SALT2 knockout animals. So what the reason of that was because it was affecting the regulation of pro-apoptotic genes, BAX and NOXA specifically. And here I just show a few data in relation to the NOXA promoter that is transcriptionally activated in a SALT2 dependent doses, and then we did EMSA studies for in vitro binding and chromatin immunoprecipitation experiments in the presence of doxorubicin also indicate that SALT2 bind to the NOXA promoter in the presence of doxorubicina, which was not happening in an unrelated region. And here is just a picture of the NOXA promoter, and we see here the binding sign of SALT2 that correspond to a proximal binding start codon. So the model for SALT2-dependent apoptotic response, we have that under genotoxic stress induced by doxorubicin or gamma radiation, SALT2 levels increase, and the increase in SALT2 correlate with the binding of SALT2 into the promoters of pro-apoptotic genes such as VAX and NOXA, specifically to GC-rich regions, and this is going to increase the apoptotic response. The main people involved in this work was David Escobar, Carlos Farcas, Matias Hepp, and Marcela Morales. We also have evidence that SALT2 negatively regulates cell proliferation, and loss of SALT2 associated with cancer-related phenotypes. So as you can see here in green, we see the proliferation of SALT2 deficient cells is faster than the SALT2 wild-type cells. We then immortalize our primary cells with the large T antigen from the simian virus, and we study cancer-related properties such as loss of contact inhibition for the focus formation assay, and you can see here the SALT2 deficient cells have an increased number of foci compared to the wild-type cells, and we also measure anchor independence growth by the esophageal assay. Again, we noticed that SALT2 immortal cells were able to have an increased number of colony compared to immortal SALT2 wild-type cells or the primary cells. In relation to the effect of SALT2 in cell proliferation, we evaluated cell cycle analysis, and we synchronized the cells with nocodazole and then released them into the cell cycle, and we noticed that SALT2 deficient cells have an early entrance to G1S phase. This correlates with changes in cyclin expression, specifically cyclin D1 and E1 were upregulated compared to the wild-type conditions, and similar to what we study for pro-apoptotic gene, we evaluate transcriptional regulation of the cyclins by SALT2. This is just an example for the promoter of cyclin E1, where we see that ectopic expression of either E1 isoform, that is the canonical, or the alternative SALT2 E1A is repressing the promoter activity. And here we have chromatin immunoprecipitation assay indicating that both isoforms SALT2 can bind to the proximal region of the promoter of cyclin E1. We compare also, or we analyzed data from different types of cancer, and we found an inverse correlation in several cancers. Here I just show the data from breast cancer, the inverse correlation between cyclin E1 and SALT2 expression, but we also found similar results in lung cancer and colon cancer. So the model that we have for SALT2-dependent regulation of the cell cycle indicate that at the end of mitosis, SALT2 expression increase, and the increase of SALT2 correlate with the regulation of several genes. SALT2 can induce the expression of cell cycle inhibitors such as P16 and P21, while it repress cyclin E, cyclin D, and also other people have shown that SALT2 is able to repress MYC in a way that indirectly also is going to affect the expression of P21 as well as cyclins. So as a summary of our findings in the SALT2 tumor suppressor functions, we noticed that SALT2 acts similar to P53 in the sense that can induce cell cycle arrest and apoptosis under genotoxic stress by inducing and repressing specific genes that I just mentioned. We are currently also investigating the role of SALT2 in the regulation of cell migration by collaboration with Dr. Vicente Torres, and here we have some interesting data that I expect to show you maybe for the next symposium. So we have in the context of migration like a dual role depending on the cell type. And something that I didn't show you, but also relate to the role of SALT2 as a tumor suppressor, relate to his regulation by the oncogene casein kinase 2. We have evidence that casein kinase 2 can phosphorylate SALT2 in a specific site near the DNA binding motif of SALT2, and SALT2 phosphorylation is a signal for SAL2 ubiquitinylation and degradation. So it's a negative regulation of SAL2 by CK2. And we have been testing the use of CX4945, that is an inhibitor of CK2, and we have seen that in several cancer cell lines, in the presence of the inhibitor, SAL2 expression is restored. So this is important because we can establish the tumor suppressor function of SAL2. So we have this kind of two different scenarios. In one hand, we have SAL2 as a tumor suppressor, where we know SAL2 can affect the expression of different genes binding to the specific binding site that is rich. And in the other hand, we have the data from the glioblastoma or glioma context, where SAL2 is acting as an oncogene, promoting stem-like phenotype. Something that nobody has taken too much care about is in the context of SAL2 isoforms. So we try to approach this and we further investigate in relation information about annotation of isoforms, but current information, and analyze differential expression of the isoforms using the IsoExpresso database. And we also combine this with several ChIP-seq experiments and analysis, as I'm going to show you next. The work mainly was done by Carlos Farkas and Matthias Hepp. And as I mentioned earlier, it was found that SAL2 has 3 exons and it can encode for 2 different isoforms. Current data indicate that actually there is 7 protein coding transcript annotated in ensemble, and there is additional exons. However, analysis of all these isoforms across tissues show that the major isoforms that are involved are alternative isoform E1A, a long isoform, and a short isoform. Both of them are under the control of the P2 promoter. The other isoforms were not much expressed across tissues. So this figure just shows the expression of this, the canonical isoform, the E1A long isoform, and the short E1A isoform between normal and tumor tissues. And as you can see here, the isoforms that have differential expression mainly are E1A and the short E1A, not much involvement of the canonical isoform. And the new thing, especially, was in relation to the short E1A that has never been described before. So this became for data from spliced RNA data from TCGA analyzing more than 10,000 samples in 30 cancer types. But short E1A isoform actually is mostly cytoplasmics. We expressed the short isoform and did confocal analysis and also to cellular fractionation, and we noticed that it's actually cytoplasmic, so we don't think there is too much involvement in the transcriptional response, but evidence from other transcription factors that has short isoforms, a possibility is that this isoform can act as a dominant negative. In relation to the ChIP experiments, we use available data from 2 experiments. The one is the From ENCODE project phase 3. That digging into this experiment, we realized that it was done with a fusion protein of EGFP SAL2, and the SAL2 in this fusion protein actually was the short E1A. And we analyzed the data from the Subas paper in the glioblastoma context, but the identity of the SAL2 isoform, it was not defined, it's unclear in this project. And we have our own data using HEC293 cells. Why we use this line? Because these cells express equal amount of E1 and E1A, as we detected by RT-PCR analysis using a specific primer set. And we generate additional models. So we have a ChIP-seq when both isoforms are present. We have a ChIP-seq when just E1 is expressed, because we're using CRISPR-Cas9, we delete exon, alternative exon 1A. And we have the double knockout without SAL2 that we use as a control. We were unable to get just the E1A form. The analysis was about peak distribution, gene ontology, DNA binding motif, and transcription factor network. And again, here is the people that mostly was involved in all this experimental approach and analysis. Carlos Matias and Carlos Alwin from Oregon National Primary Research Center also help us with all the analysis of the data. Just going to show a little bit about the information about gene ontology, DNA binding motif, and transcription factor network. So this is the overlap between all these different sheep experiments. Here we have the HEC contact, cellular contact, and here we have the glioblastoma contact. We notice a large overlap in the genes between the wild type HEC cells that has both isoforms and the one that has just the E1 isoform, which was expected. We didn't have too much overlap with the data from the ENCODE project that is IPs for the short SAL2 isoform, and it was just 0.1% identity between the target genes. And in the context of glioblastoma and HEC, two different cellular context, we have actually a large overlap that was 50%. Geoanalysis, functional analysis suggests that there is common targets between glioblastoma and HEC, and these targets relate to neural function, cell development, cell addition, cell migration, cell signaling, and signal transduction. So many of them relate to the known function of SAL2, and especially in the neural context. Short E1A was completely unrelated, and it relates more to chromatin organization and cell communication. In relation to transcription factor network, something that was interesting is that independent on the cell context, glioblastoma versus HEC293, that are embryonic kidney cells, we noticed that SAL2 is in a network with PAS and PUF, which is conserved. And interestingly, when we have just more related to E1A isoform, SAL3 was added to this network, and when just present E1, GLI3 was added to the network. Another interesting thing relates to the presence of the SOX2 in the transcription factor network. Previously, Suva demonstrated that when he found the 4 key neurodevelopmental transcription factors able to differentiate the glioblastoma cells into stem-like type, SOX is in this network. We confirmed that, and we also found that in our studies, actually SOX2 was not present in the HEC293. We did analysis, Western blot analysis, and SOX2 was not related. So we think that in this context, there is an important relationship between SAL2 and NOX. Suva also showed in his paper that actually there is a physical interaction between SOX and SAL2, as he demonstrated by co-immunoprecipitation. So in this context, SAL2 is able to bind DNA, but not into the canonical consensus site GC-rich motif, but into AT-rich motif that actually are more related with the SOX-like binding sites. Yeah, but in the context of HEC, when both isoforms are present, SAL2 is binding to the GC-rich motif, and when just E1 is present but not E1A, it is binding to a different motif, that is CT-rich motif. And so what we believe, because here we notice that the data from the E1, we notice that the number of genes that are potentially regulated by SAL2 is much, much larger than when we have both isoforms. So we think that E1A somehow is controlling or repressing the specificity for the binding, and it's more unspecific here. So the take-home message of this presentation, for you to remember that SAL2 transcription factor is altered in several cancers, and this relates mainly to the large E1A isoform and a short E1A isoform. SAL2 is upregulated in the glioma context and is essential for the maintaining of the stem-like properties, acting as an oncogene. In this context, mostly we think is the contribution from E1A isoform bind to AT-rich sequence. And we believe that this is in a SOX2 dependent manner. In the context of a tumor suppressor, SALT2 is a tumor suppressor in ovarian cancer and in normal embryonic fibroblasts, as we show from our data. And in this context, the function of SALT2 in the inhibition of cell proliferation and induction of apoptosis relate to the binding of SALT2 to GC-rich sequence to specific targets. Independent on the cell context, SALT2 works within a network of transcription factors that include PUF3F and NPAS, and depending on the tissue-specific or isoform that is present, could also be part of a network with SALT3 and SOX. Now, the significance of the SHORI1A in cancer remains to be determined. So, acknowledgement, we have collaborators from Universidad de Concepción, we have collaborators from Universidad de Chile, and we have international collaborators that help us with different models and some of the techniques that we have been using during these years. This is the current crew of our lab members. Many of them are undergrad students and graduate students from the Universidad de Concepción. And here is a special thanks to the funding and all the collaborations we have. And just this final picture of the beautiful mural of the Pinacoteca in the House of Art in our university was done by the artist, Mexican artist Jorge González in 1965. So whenever the pandemic is over, I invite you here to the university to see it in person. So thank you very much.
Okay, Roxana, thank you very much for your presentation. As I mentioned at the onset, questions can be asked at the end. Actually, the best is if you jot down your questions in the chat, and so we will address them in the roundtable discussion. We're running a bit late, but that is in part because yesterday it was still unclear whether we were actually going to have the keynote lecturer with us or not. So I think people extended their presentations somewhat. So anyway, keep with us. We are now going to hear from Ariel Castro. He got his PhD from the University of Buenos Aires in Argentina.
Can you see my presentation?
Yeah, it's fine, Ariel. In Argentina in 1995, then did a postdoc at the University of Texas Medical Branch and the IUPUI in the US with Lawrence Gilliam, where he identified the small GTase Reb as a key molecular link between growth factors and the mTOR pathway. And then he moved to San Francisco and was an assistant professor there. Was assistant professor there at the University of California, San Francisco, until then 2010, when he moved to the University of Concepción, where he is now associate professor. And he will talk to us about NUAC1 kinase, metabolic reprogramming, and driving cancer resistance to therapy. Ariel, it's all yours.
Okay, thank you, Andrew, for the invitation. To start my presentation, first I would like to focus your attention on the dynamic of the growth of solid tumors. What you see here, when a malignant tumor is established, you can see that there is an heterogeneous population of cancer cells. Within the cancer, within the tumor, there are several cancer cells that are under stressful conditions. This stress can be metabolic stress, oxidative stress, and also hypoxia. In the periphery, you can see that cells are closer to the environment, then they have high pathological rates of cell growth and proliferation. And all of these cells in these tumors need to reprogram their metabolism to adapt and survive to these conditions. And because of that, they alter the bioenergetics, they enhance the biosynthesis that need to synthesize macromolecules that are to sustain proliferation. And also they need to balance the oxidative stress because this can be cytotoxic to the cells. Considering this scenario, it's important then to find molecular mechanisms that are associated with the metabolic adaptation of the cells. We know that at least there are two main kinases, the mTOR C1 and the AMP kinase, that play a central role in cellular homeostasis. mTOR is more oncogenic because induce the activation of many metabolic pathways. In the case of AMPK, is more behaving like a tumor suppressor because inhibit all the metabolic process, but also in the case of AMPK, under stress condition can be also oncogenic because can induce cancer cell survival. We have been working for many years in the signaling mechanisms that control these kinases, but now recently we focus in a member of the family of the AMPK catalytic subunit family that has 14 members. We focus specifically in NOAC1 because it's overexpressed in many cancers. This as an example, this graph shows overall survival of patients in different cancers. And you can notice here that high levels of NOAC is associated with poor prognosis. The question is how NOAC is linked to cancer. Mainly, there are more evidence suggesting that NOAC promotes cancer cell survival, migration and invasion, therefore is involved in metastasis in several types of cancer cells, and probably more related to cancer cell survival is known that now NOAC1 is associated with cell metabolism. There are two publications from the Daniel Murphy group in University of Glasgow, one that showed that NOAC maintains mitochondrial function in big derived tumors, and also, especially in colorectal tumors, NOAC1 can counteract oxidative stress. Particularly in this last publication, they have shown that NOAC is inducing the localization, the translocation of the NRF2 transcription factor to the nucleus. And NRF2 is an important transcription factor that induces many genes associated with oxidative stress. However, in this paper, they haven't shown any direct target of NOAC that can be associated with the oxidative stress. In agreement with this study, we have been characterizing the function of NOAC in the mitochondria. We have done this seahorse analysis in collaboration with Álvaro Elorza at University Andrés Bello. And what we have seen here using this specific NOAC1 inhibitor that is indicated here like HCH, we have seen mainly that NOAC is affecting the maximal respiratory capacity of these breast cancer cells, the MCF7 and the MDA breast cancer cells. And what we have seen, interesting, that inhibition of NOAC with the inhibitor, you can see here in this microscopy that compared to the control cell, we have seen these donut-like structures. The same is true with the silencing with a specific shRNA for NOAC. We don't know how NOAC is affecting the mitochondrial function, but what we have done here is probably according to the effect of NOAC in the oxidative stress, we use two oxidative stress inhibitor, in acetylcysteine and Trolox. And you can see that again, with the inhibitor, we see these donor-like structures, but it start to be reverted by these oxidative stress inhibitors. But the main question here is what is the molecular mechanisms for the NOAC1 functions, or what are the NOAC1 targets? When we start working in this kinase, it called our attention that, for instance, in this study from Lu and collaborators, this tissue from glioblastoma tissues, you can see that NOAC is more expressed in the tumor sample and is getting higher expression in advanced grade of the disease. But interesting, you can notice in this one, especially in this one, that NOAC is expressing in the cytoplasms and in the nucleus. Then we did some studies in different cancer cells in colorectal cancer cells and also in breast cancer cells. And you can notice from this Western blot that there is a dissimilar localization of NUAC, especially, for instance, in these colon cancer cells is almost complete in the nucleus, while in the breast cancer cell MDA is almost complete in the cytoplasms. So because of the size of NUAC1, we are expecting that cannot be passive translocated to the nucleus, so then should be an active transport of this kinase. And we analyzed the sequence of NUAC, and what we found in the middle of the sequence, we found a putative nuclear localization signal. And in the N-terminus, we found what is called a bipartite nuclear localization signal that has a minor and major motif. And interesting, both motifs are conserved between mouse and human. I don't have time to show the results, but when we mutate the putative domain that is in the middle of the sequence, we haven't seen any effect on the localization of NUAC. And then in this confocal microscopy, you can see these are HeLa cells. You can see that the wild-type protein is mostly in the nucleus of the cells. But if we go directly to the mutation of the major and minor motif of the bipartite mutant, you can observe that it's almost completely lost of the localization of NUAC in the nucleus. Just a summary, this is already published. A summary of our results is that NUAC is the cargo protein, and that we found that interact with importin beta-1, and also either with IPO7 and IPO9, that also these proteins are from the family of importin beta proteins. And then these proteins are transporting NUAC to the nucleus. We don't know whether the expression or differential expression of this importin might affect the localization of NUAC or the dissimilar localization that I showed you in the previous slide. But we have done some analysis of human tissues. This is from colon carcinoma, and as you can see here, there is a correlative expression between NUAC and IPO9 expression. And if you remember, as I showed you in the cancer, in the colon cancer cell, we have found that NUAC is mostly in the nucleus. And in the case of the breast cancer cell, we have the opposite. In the invasive breast cancer cell, we find a negative correlation. Then it's probably the expression of different I-proteins, that a differential expression of this I-protein in cancer tissue may be associated with a dissimilar localization of NUAC between the cytoplasms and the nucleus. Now going to the search for the NUAC1 interacting proteins, we have done this assay, the multidimensional protein identification technology that is known as MATPIT. In this assay, we did an immunoprecipitation of NUAC. We immunoprecipitate NUAC from the cytosolic and nuclear fraction independently because of his localization in these compartments. And then after the immunoprecipitation, we digest the protein, and the peptide mixture is go to a multidimensional chromatography, and then the identification by mass spectrometry. So what we found is that in the cytosolic fraction, we found that NUAC interact with protein associated with glucose metabolism, and in the nuclear function, interact with protein associated with RNA metabolism. In this talk, I'm going to focus on the proteins that interact with NUAC in the cytosol. And mainly, I'm going to focus in the interaction with this kinase. This is the phosphofructokinase 1 that has 3 isoforms: platelet, muscle, and liver isoform. And as you can see here, they all show in the MATPIT analysis. Just to remind you where PFK1 function in the glycolysis, this is a very important enzyme because it catalyzes an irreversible reaction in the glycolysis. And I would like to notice that whenever PFK1 is inhibited, the glucose metabolized through the pentaphosphate pathway. The pentaphosphate pathway, if you remember, is important to the synthesis of ribose for the synthesis of nucleotides, but it's also very important for the generation of NADPH. NADPH is used for the fatty acid synthesis, but also it's important to counteract oxidative stress because reestablish the glutathione levels and then decrease the ROS levels in the cells. So then the question is how NOAC is affecting PFK1. So we focus mainly in this isoform, the PFK1P, because this is the one that is mostly overexpressed in cancer. So first, we need to validate the interaction with these co-immunoprecipitation studies with endogenous proteins immunoprecipitating PFKP, and then see the co-interaction with NOAC. In the confocal studies, you remember in the MDS cells, NOAC is mostly in the cytoplasm. We can see that there is a colocalization between these two proteins. But to confirm that, in fact, NOAC is affecting the interaction with PFKP in the cytoplasms, we use this mutant that I showed you before. This mutant is cytosolic mutant, and we can see the immunoprecipitation of this mutant co-immunoprecipitate with PFKP, indicating that in fact this protein are interacting in the cytoplasm. And this interaction between endogenous protein was confirmed also in breast cancer cell and in the colon cancer cells, DLD1 cells. Then we tried to analyze how NOAC was affecting the PFK1 activity. We overexpress NOAC wild type, and we see a relative decrease in the activity of PFK1 that was blocked by the specific NOAC1 inhibitor using a different approach, silencing NOAC that you can see here. We have the opposite results. In this case, when we decrease the expression of NOAC, we have an increase in the relative activity of PFK1 activity. And the same is true with the specific inhibitor of NOAC. Then what we are expecting then, if NOAC is inhibiting the activity of PFKP, so we should expect that the glucose should go through the pentaphosphate pathway. The best way to study this change in the metabolism of glucose is to do metabolomics. We have arranged some collaboration with Daniel Murphy from the University of Glasgow. Unfortunately, because of the pandemic situation, this studies has been delayed. But we have used, in the meantime, other approach to see whether NOAC is affecting the or is inducing the pentaphosphate pathway. We measure NADPH levels and ROS levels. Before going to those studies, I would like to mention that in the case of CDKC, it has been recently published that in conjunction with the cyclin D3, CDKC can can inhibit the PFK1 activity, and is doing that through phosphorylation, to specifically phosphorylation PFK1. And in fact, this inhibition of PFK1, as I told you before, it induced the metabolization of glucose to the pentaphosphate pathway, and they demonstrate that through this mechanism, CDKC can inhibit the oxidative stress and then induce the cancer cell survival. So what we're expecting is that NOAC probably is working the same way, probably because NOAC1 is a kinase, probably can induce the phosphorylation of PFK1. We have done some bioinformatic analysis, and we have identified threonine that is conserved in all the PFK1 isoforms. So the next experiment will be to do some in vitro kinase assay and some mass spec to see whether in fact NOAC can phosphorylate PFK1. So going to do the experiment, trying to see whether NOAC can affect the NADPH levels. So if NOAC is inhibiting PFK1 activity, it should increase the NADPH levels. In this case, we use the specific inhibitor, and we can see that in both breast cancer cells and the MDA and the MCF cells, we have a significant decrease in the levels of NADPH, suggesting that the inhibition of NOAC will induce glycolysis instead of the pentaphosphate pathway. And interesting, there is a commercial drug whose primary targets are NOAC and CDK4 and CDK6. We preliminary test the effect of this drug compared to the NOAC inhibitor. Here again, you can see that NOAC can inhibit the relative NAT pH levels in the cells, and we have a stronger effect with the double inhibitor. Going to the ROS levels, we are expecting here that now the oxidative stress should increase. We did some flow cytometry studies, and with the HTA inhibitor, as expected, we see an increase in the ROS levels. And with the double inhibitor, we can see a more substantial effect in the levels of NAT pH, of ROS, sorry. So it has, probably this study might have some potential therapeutic implications. In the case of CDKC, mainly because of the effect of CDKC in the cell cycle, there are inhibitors such as palbociclib. Palbociclib has been approved for advanced or metastatic breast cancer. It's also in phase 3 for metastatic colon cancer. Unfortunately, as usual for this kind of inhibitors, there is resistance to the inhibitors. Here is a summary of some of the mechanisms for the resistance to palbociclib. Mainly is the activation of CDK2 that can bypass the CDK4 and CDK6 for the cell cycle progression, and also there is activation of the PI3 kinase signaling. Because of that, in the clinic, they have been used some combined therapy with the palbociclib and some PI3 kinase inhibitors of CDK2 inhibitors. Because of the similar pathway that we think NOAC is affecting, in addition to the pathway that CDKC is affecting the oxidative stress, we think that maybe NOAC is driving some resistance also to palbociclib. We initially did some experiments to see what happened with NOAC expression, and you can see when you incubate these breast cancer cells with different concentration of palbociclib, you see a significant increase in the expression of NOAC in both cancer cell lines. The same with the colon cancer cell, colorectal cancer cells. And apparently this effect is due to a transcriptional effect, because we did some real time, and in all the same cancer cells, we can see that the incubation with palbociclib induced an increase in the expression of mRNA levels of NOAC. To see whether there was an effect in cell survival, we initially used cleavable as an indicator of apoptosis. In this case, we compare the effect of the NOAC inhibitor, the double inhibitor, and palbociclib. And it is clear here that the double inhibitor induced a significant increase in cleavable PARG, indicating that cells are going to apoptosis. And is, of course, is very efficient compared to the other two inhibitors. And the same in these breast cancer cells, indicating that then the combined inhibition of NOAC and CDK6 might be more efficient to induce cancer cell death. So as a summary of the result that I shall show you, our model is NOAC1, that is, I show you that has two locations in the cytoplasm and in the nucleus. The cytosolic NOAC will be inhibiting the activity of PFK1, probably through phosphorylation. The inhibition then of PFK1 will induce the pentaphosphate pathway, and this pathway, through the increase of NAT pH level, we are expecting that decrease the ROS levels and levels in the cells, and of course the potential cytotoxic effects of ROS, and then through this pathway now is affecting the cancer cell survival. Just an acknowledgement, this is a picture of our group that we share with Roxana, my wife, and Emilia has done most of the experiment for the characterization of the mitochondria. Mario has been involved in the interaction of PFK1 with NUAC, with the MATPID analysis that we have done with in collaboration with Sherry Workman in the Stower Institute. And here as our collaborator, University of Concepcion, in the Stower Institute, and also Universidad de Bello, Álvaro Lorza for the mitochondrial studies. And that's it. Thank you very much. And the funding, of course.
Okay, Ariel, thank you very much for your presentation. With that, we can open the round table discussion session. We are a bit behind time, but I think we still have about 10 minutes we could use for questions. So I will proceed to the chat and to see what we have here. So for instance, Lisette Layton asks Vicente Torres, how does a migrating cell know whether active RAB5 has to activate RAC1, as was shown in my presentation, or function in the endocytic pathway? Is there any kind of sort of molecular switch there that tells RAB5 what precisely to do?
No, so far it remains unknowing on whether RAB5 switches between a rather mechanical focal adhesion disassembly or as acting as a signaling platform in the early endosome. So what is known is that RAB5 triggers the activation of RAB1 from the early endosomes by recruiting the GEF-TIAM. But on the other hand, RAB5 promotes the disassembly of focal adhesions by activating this protease calpain in early endosomes. But it's not knowing how does it switches between one or the other mechanism.
Okay. Oh, yeah. I had a question there related to the recruitment of calpains to the tail end of cells there, and participation in the degradation of the focal adhesion complexes. So how exactly does the RAB5 recruit the protease? Is the mechanism known?
It remains unknown, but former reports previous to our studies showed that calpain 2, along with calpain 1 to a lesser extent, are accumulated at early endosomes and endocytic vesicles that fuse with early endosomes. What RAB5 is doing once it's activated is promoting the relocalization of early endosomes loaded with calpain 2 to the cell periphery. And when you knock down RAB5, these early endosomes are relocalized, are redistributed, and calpain 2 is also decreased from these early endosomes, but the mechanism is not known yet. We suspect that it might be involving an intermediate protein, calpastatin, but we have very preliminary data in that respect, and we cannot be sure about that.
Okay, thank you, Vicente. There is a question here by Sergio Lavandero for Fabiola. Could all speakers activate their microphones and also the video so we can see you? So it says here, in our hands, in vivo chronic administration of L-name, so it's a NOS inhibitor, generates nitrosative stress by inducing iNOS and nitrosylation of some specific protein. So it sort of would induce the opposite effect. Do you have any comment there?
Yes, all the effects caused by nitric oxide depend strongly on the nitric oxide concentration. So you may have opposite effects depending on nitric oxide concentration. And INOS, which is the enzyme that Sergio is talking about, is characterized for inducing very high amounts of nitric oxide. So it's not weird that it's causing the opposite effect.
Okay, thank you. So there's another question here for Fabiola. At which time frame, or in which time frame, do pro-inflammatory cytokines increase endothelial permeability? Have you ever tested CD90, so Thi1, as an endothelial cell adhesion protein, which increases expression upon TNF treatment, so which is what you were using in your experiments, and CD90 has been shown to bind to alpha V beta 3 integrin in leukocytes and in cancer cells, and helps them then to transmigrate the endothelium.
Okay, well, for the first question, the minimal time that we have seen permeability is in 15 minutes for the way that the experiment is done. However, INOS gets activated at 1 minute of application, but in 15 minutes, the permeability occurs physiological more faster, but we can measure in just 10 minutes is the minimal that we can measure. And the other question, CD90, we haven't tested that protein, but we are open to test it. If Lisette sends us the antibody, we can test if increases in response to our pro-inflammatory stimulation, and if the mechanism is by clustering or traffic, and if it is regulated by S-nitrosylation. Yes, we can do that.
Well, great. So the idea, of course, of these talks is to foster interaction. So hopefully some collaborations will come out of this discussion. I have a question here for Roxana. So I was wondering whether the short E1A acts as a dominant negative for the longer isoform, perhaps by preventing dimerization or something like that?
Hi, Andrew. Actually, we don't know. We haven't tested yet, but definitely the experience from other transcription factors that have isoforms, short isoform present in the cytoplasm, this is one of the mechanisms. They are as a dominant negative. They cannot bind DNA, but they can interact, for example, with part of the complex co-regulators. And in this way, they can form inactive complexes, so they can change the activity. We want to test that as soon as possible, and it's easy to do because we have all the tools to do it, but unfortunately now we cannot go to the lab. But there is this possibility, definitely.
Okay, fantastic. Vicente Torres has a question for Ariel. And he's wondering whether NUAC1 subcellular localization changes in hypoxia. Can you comment on that?
Yeah, we don't know. We know that NUAC1 subcellular localization change with the oxidative stress and also with low glucose. And there is a paper that showed that during hypoxia, the expression of NUAC increase. But we haven't tested that. It's a very good question. I don't know.
Okay. There have been a couple of questions for me here too. So Roxana was wondering about caveolin regulation at earlier stages of cancer versus metastasis. What is responsible for loss and gain of function, respectively? So with respect to the regulation at early stages, the downregulation, is thought to be by epigenetic mechanisms, so silencing of the promoter region by methylation. That's fairly well documented. And then obviously this methylation can be reverted later on. And in terms of what is responsible for the changes in function, well, essentially we propose that E-cadherin plays a key role in that respect, in that in cells where caveolin is expressed and E-cadherin is present, it will have this tumor suppressor function, but in cells where caveolin would be expressed in the absence of E-cadherin, which would be in late stages of cancer, it would have this opposite role, because it would then connect to the signaling pathway I mentioned there, which is via RAB5 signaling and then RAC1 promoting their activity. And this would not occur in E-cadherin expressing cells. There was also then a question concerning whether caveolin-1 plays a role in sorting of molecules into exosomes, proteins or RNA samples. This was by Eduardo Durante. And the response there is yes, at least in terms of the protein, determining the protein composition of exosomes. Caveolin does seem to play an important role, and to do so in a phosphorylation dependent manner. So the phosphorylation of caveolin is not only relevant to the activation of this RAB5, RAC1 signaling cascade, but it also will determine what kind of proteins are get incorporated. So I didn't show you the results, but this is something we're working on now to show that actually the exosome composition differs depending on whether you have a phosphorylated or a non-phosphorylated caveolin. So caveolin promotes the targeting of specific proteins to exosomes, or the inclusion of specific proteins in exosomes, and does so in a phosphorylation dependent manner. So with this information we obtained by obviously doing a mass spec analysis with our collaborator in the US, Jay Heineke. There's a question here for Vicente from Denise Bravo, and she's wondering whether oral bacteria can activate changes associated with dysplasia.
Interesting question. However, I don't have any answer yet to that, because concerning oral dysplasia, very little is known about the contributors to the onset of this. What is known that the relationship between alcohol and intake and smoking affects this the onset of oral dysplasia, but concerning the microorganisms is not very clear yet.
Okay, thank you. Then there's a question here by Ariel, and that is alsin-2 only activated by increasing its expression during hypoxia, or does it also have a regulatory domain that could link to its activation during hypoxia?
Another interesting question without a positive answer. We basically, I can see here in the people Sole Rivas, which performed the studies, is basically we observe changes in the expression at the mRNA level and protein level, but not activation of alsin-2 is not well known. The mechanisms of regulation of alsin-2, it's known to contain pH domains that associate with lipids, but it's a very poorly studied protein so far.
Okay. Looking here. There's another one here for Vicente. So could saliva and its components play a role in the migration of oral cells, for instance, exosomes from saliva?
Sorry again, I had problems with that.
Could saliva and its components play a role in the migration of oral cells, for instance, exosomes from saliva?
Oh, Wow, this is a field that we are researching in the lab. I didn't talk about that here, but we know that components of saliva promote cell migration, and this is mediated by a similar mechanism deregulated in tumor cells. That is the activation of RAP5. We indeed showed that components of the saliva stimulate migration of epithelial cells by activating RAP5 and then downstream RAP1. So we believe that this conserved mechanism that is deregulated in cancer, enhancing cell migration, is also observed in this during wound healing process. That is reminiscent of that happening in tumor cells.
Okay, thank you. There's a question here for Fabiola by myself. I was wondering whether pre-clustering of ICAMs is absolutely necessary for tumor cell binding to endothelial cells in a static system. I would imagine you could still get binding even if you didn't have clustering, but what I'm thinking is it probably helps considerably under flow conditions. Have you actually looked at that?
We haven't done experiment in flu conditions, but it's unknown if ICAM1 clustering is necessary for tumor cell adhesion. So far, we have tested that is necessary for leukocyte adhesion, and that have tested in the literature too. But we have to test it if it's really is necessary for tumor cell adhesion. So we don't know yet. We have to do the experiment now, and I imagine that flu conditions can help very much. Yes.
So the clustering would help under flow conditions. That would be the conclusion. Yeah. Okay. Then there's another question here for Ariel, actually by myself. You're looking at the interaction between NUAC1 and PFKP, and I was wondering whether it's direct or indirect. I was thinking it's more likely to be indirect because generally protein or kinase substrate interactions are very transient, and since you were able to identify it in an IP experiment, I would think there are intermediate binding partners there. So I was wondering whether you've done any experiments along those lines.
Okay, yeah, I guess one is that is direct because we start setting the experiment for the in vitro kinase experiments. We are working with purified proteins for PFK and NUAC, and we can still see interaction. But still in the cells, probably is NUAC forming a complex with this protein, another protein from the glucose metabolism. So probably that's why we are seeing this interaction in the immunoprecipitation. So it still might be some protein that is affecting the interaction between them. But in the in vitro experiment, so for the kinase experiment, we still can see the interaction with the purified proteins.
Well, you see interaction or phosphorylation?
No, we haven't done the phosphorylation experiments. We just start doing the in vitro experiment. We see the interaction. We see the interaction. And then the experiment will be done soon.
Yeah, okay. So Marcella Kogan is asking me here about how long the effect of exosomes lasts in terms of modulating the metastatic behavior of cells. So of course, when we do the experiments in vitro, we incubate for 24 hours and do the migration assays or the invasion assay. And when we do the experiments in vivo, we actually pre-incubate the cells with the exosomes and then subsequently every 4 days inject exosomes again. Now this is a protocol that was pre-established for other reasons, and we never went back actually to test, for instance, in the in vivo system with whether one and only one pre-incubation would be sufficient to get, for instance, the pro-metastatic effect and that we see with these incubating with these extracellular vesicles. So, in short, we're not really sure how long the effect lasts. I think that almost wraps it up here. Oh, there was a question there about, yes, there was a question there by Sergio Lavandera about stating that extracellular vesicles produced by adipose tissue contain organelles such as mitochondria, and whether we had checked this possibility in our model. So mitochondria are fairly large structures. They are probably on the order of about 500 nanometers, plus minus, to a micron. And the extracellular vesicles, they come in different flavors. So you have vesicles that can be beyond micron size, but exosomes are essentially the smallest group of vesicles in the category of extracellular vesicles. And their genesis is via an endosomal pathway, via the multivesicular bodies and then subsequent fusion with the surface. So given their size, as I showed you, which is roughly 150 nanometers, 100 to 150, it's highly unlikely that entire mitochondria could be included in such structures. And indeed, in our proteomics analysis we've done with different exosome preparations, we actually were not able to detect mitochondrial components. So I think they are definitely not included in exosomes. They might be included in other types of extracellular vesicle populations that would have to be considerably larger. Okay, with that, I think we'll wrap up this session. As I said, we're a bit overdue on time. Because we were uncertain about the plenary election, so people extended their talks a bit, but that's not a problem. We'll take a short break now. In this break, you will be shown a video summarizing what the last symposium was. So the ACCDiS Cancer Spring Symposium #1 in 2016. There were some photos of that for entertainment. And then let's say at 10:45, we'll be back again, and we'll be getting ready to hear from our keynote speaker, Luis Carvajal. Okay, so take a short break, grab a coffee, and look at the video. See you in a moment.
grito te doy mi vida. ¿Para qué? Mi vida, para que no llore.
Hello? Hello?
Hello, Luis.
Hello, hello Alejandro, ¿qué tal?
Hello, this is Alejandro from Chile. Well,
Hello,
thanks for joining us. Okay, let me introduce for our plenary lecture Dr. Luis Carvajal, who obtained his PhD from University College of London in 2003, and then carried a postdoctoral training at Cancer Research UK London Research Institute. I was a senior research fellow at the University of Oxford. After that, he took a position at the University of California in Davis, where he's currently Associate Director for Basic Science and Co-Director of Community Program Clinical Translational Science Centers. Dr. Carvajal research interest is human genetics at population, clinical, and laboratory levels. And he has co-authored more than 80 high-profile publications. So for me, it's a pleasure to introduce Dr. Carmona, who's also a collaborator in our projects, with his topic about recent advances in epidemiology, genetics, and genomics of gastric cancer. Dr. Carvajal.
Okay, thank you very much for inviting me. It's a pleasure to be here. I have a lot of collaborators in Chile, and I enjoy working with all of you. So let me share my screen now. Okay. Okay, so can you all see my screen?
Yes.
Hello?
Perfect.
Okay, good. All right. Okay. So, thank you very much for inviting me to present here. I am Luis Carvajal. I work here in California. It's early here. And I have been working lately on stomach or gastric cancer. Today I am going to cover three main areas. One is the epidemiology of the disease. Then I will give you some information about genetic susceptibility. And finally, I will discuss some of the more recent work on somatic genetics and heterogeneity in this common disease. So let's start with gastric cancer epidemiology and disparities. So gastric cancer is an important cancer worldwide. It's the third cause of cancer death. There are around 1 million new cases every year and around 730,000 deaths. As you can see, there is no disparity between incidence and mortality. It's a disease with a really poor prognosis in general. And it's mostly because it's very difficult to detect when it's in early stages. And when it's detected, which is mostly metastatic disease, the survival is very low, around 5%, the 5-year survival for metastatic disease. Here in the US, is the leading disparity in Latinos or Hispanics. And there are only two targeted therapies approved by FDA. More recently, immunotherapy also was approved for gastric cancer. So difficult to detect, and when it's detected, there are not many options to treat the patients. There are multiple risk factors for these conditions. It's more common in men, it's more common in Latinos, as I said. The highest incidence is in Asia and South America. And the main risk factor worldwide is Helicobacter pylori, a bacteria that colonizes the stomach. Also, smoking is a risk factor, as it is Epstein-Barr virus infection. Is a disease that is more common in the old age. The average age of diagnosis here in the US is around 70. There are other also risk factors in the diet, and reflux or GERD is also a risk factor. And 1 to 3% of all patients have inherited diseases, mostly hereditary diffuse gastric cancer, although we discover a new form which involve other genes in DNA repair. However, around 15 to 20% of all patients report relatives with cancer. So the role of genetics in disease may be higher than anticipated. Also, the epidemiology depends on where the tumor is located. So the upper tumor, the tumors of the cardia of this part of the stomach, they have a different epidemiology to the non-cardia tumors, which are in the lower part of the stomach. For cardiac tumors, being white, having reflux, and being a man, all those three are risk factors. And in fact, here in the US, one of the few groups where the gastric cancer incidence is increasing is in white men because they have high levels of obesity. For non-cardia, the most important risk factor is being infected with this bacteria, Helicobacter pylori. And we talk here about disparities, which is very, it's a good common terminology here in the US. Here I'm showing you the incidence and mortality of gastric cancer in Hispanics, in whites and in Hispanics, and data from Mexico and Colombia. And as you can see, compared to whites, the incidence is higher in both in men and in women, as it is the mortality. So it is one of those cancers that here the National Cancer Institute is really interested in supporting because of this higher effect in this minority population. And of course, for us in Latin America, it's a really important condition too. There are interactions that have been identified lately. For instance, there is a relationship between location of the tumor and race and ethnicity. This is a paper published in Gastroenterology where they discovered that for cardiac cancer, the incidence is going up in men, but it's coming down, is decreasing in minorities. And there is no association with socioeconomic status, okay? For non-cardiac cancer, the incidence is increasing in minorities, but decreasing in whites, and has a strong association with socioeconomic status. So basically, non-cardiac cancers tend to become more common in minorities, particularly in poorer minorities. This is another example of a study that we published here in California, where we compare the epidemiology of gastric cancer in whites, blacks, Hispanics, and Asian Pacific Islanders. So just to highlight, this is a big busy table. Something that we found is that Hispanics have a high incidence in young people. Around 23% of the cases here in California are diagnosed in people under 50. It's more common in the lowest socioeconomic status. And there is also something interesting that we notice is there is the fraction of tumors with diffuse histology. It was surprisingly high. It's around 33%. And so that histology seems to be increasing in incidence here in California. So clearly, the take-home message for this busy table is that the epidemiology of gastric cancer is different across races and ethnicities here in the US, because they probably have a different risk factor profile. Okay. Okay. Something else that is interesting here in the US is that even within racial groups, there is a lot of variation. And in the US, as Latinos or Hispanics are really diverse. So here in the West Coast in California, we mostly have Mexican Americans. In the East Coast, we have mostly people from the Caribbean and South America, Cubans, Puerto Ricans, and they have a different risk factor profile. The same happened with Asians. So in here we have in the US we have multiple Asian populations. And there is quite a lot of diversity in these groups. And here, this study demonstrated that the group with the highest survival rate of these Asian groups is Koreans. And probably because in Korea, a lot of them are used to the national gastric screening program they have in that country. So probably they are more aware of the symptoms, and they probably seek to endoscopy more often than other groups. So that's interesting. So I will say, as this paper is called, one size doesn't fit all. I wouldn't say, when I say for Latinos, it's a broad generalization. I'm sure there are differences within Latino populations. This is another interesting study were published by a group in the NCI, where they noticed that even in whites, there are some interesting patterns, particularly it seems like the incidence is going up in young white women, in kind of middle-aged white women. That is something that is not well understood. It's probably related to the increasing atrophic of autoimmune gastritis. But it's a recent finding, and we don't understand much of what's going on there. And this is another example of how the disease is different in whites and non-whites. As you can see here, non-cardiac cancer is more common in minorities, but cardiac cancer is increasing in whites. We are also quite interested in gastric cancer because of another risk factor is obesity. Okay, so here in the US, I don't know much details in Chile, but in general in South America, obesity has been increasing. So there has been a trend also in cancer incidence. So here in the US, nearly all 12 obesity-related cancers, which are shown in this table, are increasing in incidence in young people. Okay, so here I'm showing these are different cancer types, and this line separates and the age of onset. So on the left are early onset tumors, and on the right are late onset tumors. And these are the annual changes in incidence. So as you can see, most of these obesity-related cancers are increasing in incidence in young people. It's a good example for colon, for endometrial, and also for cardiac cancer. So we have been trying to disentangle this relationship here, but at least for gastric cancer, for cardiac cancer, and also for the closely related esophageal cancer, it is possible that that the recent changes are driven mostly by the obesity epidemic that is impacting mostly people born in the 80s, 70s, and 80s. This is also a comparison of the same figure that I was showing you, but by racial group. So here I'm comparing whites and blacks. And as you can see, there are important differences. In whites, for instance, esophagus and cardiac cancer are increasing a bit more than in blacks. So then again, you know, we are trying to disentangle the effect of this obesity epidemic across multiple racial or ethnic groups in the US. This is data from the registry from the US, the SEER registries run by the National Cancer Institute. And we have a number of studies, epidemiological studies in Latin America, mostly in collaboration with people in Chile, in Colombia, and in Mexico. We were funded this year to pilot the establishment of high-risk cohorts in Colombia, Chile, and Colombia, Mexico, and the US. So basically, we are starting to recruit participants with atrophic gastritis and intestinal metaplasia. And also, we are starting a high-risk cohort of first-degree relatives of patients with gastric cancer. And we are going to do some experiments in that. So the idea is to pilot to establish that cohort. We are also characterizing the patterns of epidemiology in premalignant lesions, mostly people with, atrophic gastritis, intestinal metaplasia, and dysplasia. And also we are collaborating with, well, this is actually mostly work led by Dr. Javier Torres, who is doing a lot of work on Helicobacter pylori in Mexico and also helping us to do the same in Colombia. So Helicobacter pylori, I mentioned you probably know more about this than me, but Helicobacter pylori is the main risk factor for gastric cancer in the planet. It's a type 1 carcinogen, and the issue is that nearly 9 in 10 or more than 9 in 10 patients with gastric cancer have a history of infection with this condition. So it is actually a really important disease, infection. But also it's very common. So the issue with the Helicobacter pylori is, at least depending on where you look, in which country you look, there are, for instance, in Russia and even in certain parts of South America, a lot of people have this infection, and not all of them are going to have gastric cancer. So even though it's a common condition or a common infection, the incidence does not fully correlate with the incidence of gastric cancer. So I'm showing here some examples where Helicobacter pylori is very prevalent, but the incidence is low, like North Africa or India. This is another example in Northern South America, actually including Chile too, where there is a good overlap between Helicobacter pylori prevalence and gastric cancer incidence. And this is actually work mostly done by my collaborator, Dr. Torres in Mexico, Javier Torres, where clearly Helicobacter pylori has been evolving with human populations. And there are a number of haplotypes or genotypes that are characteristics of the main continental populations in the planet, which they correlate with race. So there are strains that correlate with Europeans, strains that correlate with Africans, Asians, and with Native American or indigenous American people. And there is also a great deal of variation in the genetic structure of these strains. Here are this data from Javier, and there are multiple strains. This is a phylogenetic tree, and there is a lot of variation, and they tend to cluster. So these are samples from Colombia, other from Latin America, and these are from Mexico. As you can see, even though these are closely related countries, they fall in different parts of the tree. So there is variation even within countries in terms of genetic diversity of Helicobacter pylori. This is another example of data from Helicobacter pylori. This is a structure plot, and as you can see, Honduras and Colombia, they tend to have different types of Helicobacter pylori when you look really deep into their genome. So this is something that we are wanting to investigate in detail. I'm sure also there are some groups doing great work in Chile about this, and I think some of you are part of this international Helicobacter pylori consortium. I'm not. Javier is part of that, but that is actually providing some interesting data. This is data from Javier and me and our group in Colombia, where we are comparing the genetic variations in patients with preneoplasia. We are defining preneoplasia here as people with atrophic gastritis or with intestinal metaplasia. And these are the different genetic contributions of Amerindians, Europeans, and Africans in patients with preneoplasia and in patients with gastric cancer from the two countries. And as you can see, there is a higher fraction of American Indian ancestry in these individuals. So we are trying to follow this in more detail, but the evidence is suggesting that American Indian ancestry, high American Indian ancestry, could represent a risk factor for gastric cancer, at least in these two countries. Okay, so that's kind of the summary of the epidemiology. There have been some new advances on epidemiology, mostly looking at different racial or ethnic groups here in the US. And this is important because there is a renewed interest to investigate this condition, particularly in minorities, such as Latinos, which are the majority in Latin America. Okay, so now I'm going to switch gears a bit, and I'm going to tell you some information about the genetics of gastric cancer. So gastric cancer has been, because we have this risk factor with such a huge effect, which is Helicobacter pylori, gastric cancer has been considered morely an environmentally driven disease or a non-genetic driven disease. However, there is, there have been some changes in the recent years. We discovered a new form of inherited gastric cancer in 2017. This is a paper where we have a lot of colleagues and friends from Chile here in this paper. We actually studied some patients from Chile here. And we found that a high prevalence of mutations in these three genes, PALB2, BRCA1, and RAF51C. And those genes are important cancer genes. They closely interact to repair DNA breaks. But until this study hadn't been associated with gastric cancer, those genes had been previously associated with other cancers, primarily breast cancer. So in this study, what we did was the results of an exome sequencing study. And in this study, we look at patients that were familial. They don't have any of the known gastric cancer genes, so we ended up finding a lot of mutations, actually 11 mutations in these genes in around 300 patients. And something that was very important in this paper is that there were some concerns about the fact that these patients could be sporadic patients, that they happen to be in breast cancer families, and they carry these mutations, but they didn't have, the mutation had nothing to do with the disease. Well, actually, we look at the tumors of these patients. We sequenced 4 tumors, and we did an analysis of mutational signatures. And there is a mutational signature that is very clear in tumors that are deficient for homologous recombination repair. That signature is called BRCAness, which is the signature that results from the deficiency in the BRCA genes. And we found that these tumors all of them were deficient in homologous recombination. So not only these patients have germline mutations in the genes, but also the tumors that they had had the expected mutational signature. So this is actually a strong evidence for causality of these mutations. Since then, a lot of groups have reported mutations in these genes. This is a summary. It's actually slightly outdated now, but clearly there is good evidence for PALB2 as a new gastric cancer gene, as it is for ATM. And there are other genes that have been reported independently. Something that is quite interesting in this condition is that the previously known genetic syndrome for gastric cancer is called hereditary diffuse gastric cancer, caused by a gene called mutation in a gene called CDH1. That condition is mostly associated with diffuse histology. In our case, these genes are mutated in both intestinal diffuse and mixed histology. So this is slightly different to the previously known condition. Okay, so that is kind of an update on the genetics of these. We are currently following up on this. We are about to screen around 600 patients in an exome sequencing study to see if we identify more gastric cancer genes. And actually, we are collaborating with a number of people in Chile, including Dr. Corbalan, to see if we discover more genes for gastric cancer. Clearly, at least the recent data is suggesting that up to 1 in 10 patients carry an inherited mutation, and that's actually quite important because 1 in 10 patients for gastric cancer is 100,000 patients a year in the planet. So that could be potentially quite important. So stay tuned. Hopefully, next year we will give you some good news about new genes for gastric cancer. Okay, so the last part of my seminar is on somatic genetics, intratumor heterogeneity, and drug efficacy testing. So one issue with the gastric cancer and the poor prognosis is that this is a really complex disease. This is data from the TCGA, from this tumor genome study here in the US, an international study. And these are different cancer types that are ranked by the mutational frequency. So on this part of the figure are the tumors with the lowest mutation rate. On this part of the figure are the tumors with the highest mutation rate. So as you can see, stomach or gastric cancer is one of the tumors with the highest mutation rate. These other tumors have a high mutation rate mostly because they are driven by smoking, as is bladder cancer, lung, and the 2 lung cancer types, and by another important risk factor, which is ultraviolet radiation, UV radiation in melanoma. So the stomach cancer is one of the tumors with the highest mutation rate. Actually, on average, when we sequence the exome of a gastric tumor, on average it has 400 to 500 mutations that can be potentially interesting. So it is very difficult to distinguish which one of these mutations are drivers and which ones are passengers. And more importantly, it's very difficult to tell if you are an oncologist treating a patient, which is the best mutation to select for a targeted therapy. It's very difficult because you have many choices. An important advance in the genomics of gastric cancer was this study at TCGA, where they sequenced around 300 patients and they did a multi-omic characterizations of these tumors. And they came up with 4 different molecular subtypes. I'm showing them here. One of them is the chromosomal instability subtype, which is mostly seen in intestinal tumors with TP53 mutations and RAS activation. Then the Epstein-Barr virus tumors, they are infected with Epstein-Barr. They are highly methylated. They have a high prevalence of PIK3 and CDK2A silencing. The microsatellite stable tumor, they have microsatellite instability. They are hypermutated and they have a high frequency of MLH1 silencing. And finally, these are the genomically stable tumors, which tend to be diffuse, have high frequency of mutations in CDH1 and ROA genes, and they have issues with SILA disease. Okay, so those are the 4 different molecular subtypes, and they have different prognostic profiles. It has been shown that the ones with the best prognosis are the ones with Epstein-Barr virus, and I think a study in Chile also showed this in your country. And the genomically stable tumors are definitely the ones that have the worst prognosis. Not only they tend to respond less to chemotherapy, but also the mortality is higher in all of these patients, unfortunately. So what we did in here in my lab was to look at patterns of intratumor heterogeneity. We collected multiple biopsies from the same tumor. So in this case, we collected a biopsy here, here, and here. And as you can see, if these are different tumor cell types, every biopsy will be sampling a different tumor population. So then what we do is we sequence the biopsies and we make phylogenetic trees. And the phylogenetic trees, we kind of are building the following way. The mutations that are found in all tumors, in this case, I would say the gray mutation, is what we call clonal mutation, okay? So founder mutation. But then there will be mutations that are not seen in some biopsies, and we call those private mutations. So the clonal mutations are present in all tumor cells. The private mutations are only present in small cell populations. So see, this data from this study, in black is the study that we did, and in red is data from TCGA. We didn't see major differences, but we found a higher frequency of genomic stable tumors in our samples, or samples that were mostly from Colombia, Mexico, and California. So that was quite interesting. We are following up on this. I need to check what you reported in Chile. There's a group that reported something about this, but our evidence and also a study from the UT Southwestern in Texas is suggesting that that Latinos are enriched by the genomically stable subtype, which is associated with diffuse histology, but also with worse outcomes. So this could explain in part these disparities in mortality that we are seeing here in the US and the high mortality we see in Latin America in general. This is actually from the same study. These are the genes that were reported as drivers in the TCGA, and this is the frequency of the mutations in our study. And as you can see, we found very similar mutational frequencies. I wouldn't say very similar, but TCGA is a study of 300, or study is around 30 patients. What we saw is that because we have this ability to identify which mutations were clonal and which mutations were subclonal, we can actually do comparison like this. So in this case, for instance, P53 is a gene that is nearly always clonally mutated, while most of the other genes, ARID1A, PIK3CA, CDH1, the fraction of clonal mutation is lower. So a good fraction of the mutations that are being reported by TCGA in the so-called driver genes are actually subclonal or private, okay? Which is quite important for tumor evolution, but also quite important for treatment, because if you want to use, say, PIK3CA, if you want to use that gene as a target, there are drugs, PIK3CA or PIK3 inhibitors. If you give that inhibitor to a patient that has a subclonal or private mutation, that the response in that patient is going to be expected to be lower, because the target, the drug is not going to affect all tumor cells. is going to affect only the tumor cells that have that mutation. So what you want is to have situations like this gene, HOA ROA gene, where nearly all, or in this case, all mutations we found in the study were clonal. Okay, so it was quite interesting. We are about to submit this for publication. These are this data from the more frequently mutated genes. This is slightly updated, but as you can see, we found relatively good correspondence between our study and the TCGA. Then again, I cannot make a lot of strong assumptions here because our sample size was only 30 patients. But this is the typical example of these findings. So this is a patient where here is N is normal, and this is the tumor biopsy at 12, at 3, at 6, and at 9. As you can see, this tumor has clonal mutations in RBB4, FAT4, EYS, etc., and TP53, and have subclonal mutation in RBB4. So all these biopsies have RBB4 mutation. This one didn't have it. And then it has every biopsy has a lot of unique mutations or private mutations. So this is the issue with this tumor type is tremendously diverse. And this diversity is mostly driven by the fact that every fraction of the tumor will have different mutations. There will be a lot of private mutations sitting around. These are other examples when in this case, we didn't find any clonal mutations that we could see, but we found a lot of subclonal mutations. And it is an example on RBB2. So in this case, RBB2 is one of the genes that is used for a targeted therapy in gastric cancer. So in this case, if you give an RBB2 and HER2 inhibitor, trastuzumab, to this patient, it will kill the cells that are in this biopsy or in this biopsy, but these biopsies don't have the target. So that drug is less likely to work in that patient. In this case, these two patients, they have clonal RBB2 mutation. So the expectation is that if there is a response, there will be a good response here because all your tumor cells will have the target. These are other examples of the phylogenetic trees that we are making from the data. And as you can see, the pattern is interesting. Nearly every tumor has a different phylogeny, which is interesting and also a big challenge because it's actually more difficult to develop new therapies in this disease. This is also the prevalence of the targets in our population. We found high frequency of PIK3CA, RBB2, and KRAS, and also a high frequency of CDK and the genes that will be targeted with PARP inhibitors like ITM, BRCA2, and PALB2. And this is the fraction of when you stratify by subtype, this is the fraction of druggable genes, druggable mutations. So around half of the Epstein-Barr virus will have a clonal target. Around 80% of the MSI will have a clonal target, but you see the genomically stable, which are the ones with more difficult to treat, are the ones with less target. Around less than a quarter of them have targets. I'm going to skip this one. This is so we are another study that we are doing here is we are making organoids and patient-derived xenografts from patients with multiple cancer types, including gastric cancer, and we are testing these organoids with multiple. We are sequencing them, we are profiling them genomically, and we are treating them with multiple drugs and chemotherapies, targeted drug and chemotherapies. And the most promising ones, we are taking them forward and we are doing animal studies in PDXs. So we are actually doing a lot of that work here. This is an example of one of those drug efficacy testing. So in this case, we found this model, which we found this model. Some of these models are responding well to this CDK inhibitor called palbociclib. So we are basically investigating the genomics of gastric cancer, and we are trying to do some translational research to see if we can come up with better combinations that can be tested in clinical trials. So this is my last slide. Gastric cancer is an important condition. It's a leading cause of morbidity and mortality in all world populations, including Latin America. A good fraction of these tumors are caused by inherited conditions, which has been underappreciated. And finally, the complexity of these tumors is tremendous, is very high, and it has high implication for therapy. So these are my acknowledgement slides. We collaborate with people everywhere. I'm sorry if some of you are not here, and we are also funded by multiple grants from the National Cancer Institute. They have been very supportive of our work. Thank you very much. I'm happy to answer questions now.
Thank you, Luis. Now this conference is open for discussion. I'm checking the chat. I don't know, is there any questions to Luis? Okay, I have one initial question. What happened with P53 in terms of germline mutation, because it has been described in other tumors, and also is commonly as a somatic mutation in gastric cancer?
I think, there could be probably mutations in the genes, but I don't think the prevalence is that high that had been discovered. The genes that are showing the highest prevalence are, at the moment, PALB2 and ATM outside CDH1. The cluster of mutations seems to be more on the homologous recombination pathway. But P53 is an important gene. Is one of those tumor suppressors that is the most commonly mutated tumor suppressor gene in gastric cancer, but it seems the silencing is more somatically, not in the germline.
Okay. Any other question? Fabiola?
Yes, it's a very general question about how can we do to prevent gastric cancer, or there is something in the diet, or there is like an exam, for example, a colonoscopy that you can do, and what age do you recommend? That is very general question.
Okay, well, I think the problem with gastric cancer is that there are no good risk prediction models. Okay, so it's very difficult to tell which people in the population are at high risk. Okay, and there are some countries in Asia, Japan and Korea mostly, they have national screening programs where everyone is offered open endoscopy. Okay, I think starting at 40. Okay, those programs are extremely expensive and logistically challenging. I cannot think of any country in Latin America or even here in Europe that have the capacity to screen everyone for gastric cancer. So I think it's important in the field for us to know more about the risk factors, okay, and to test this in longitudinally in cohorts, okay, that allows to identify those groups that are at higher risk, and then come up with models that allows them to kind of prioritize them for screening. Another important element is that if we make more advances on the genetics of gastric cancer, we could offer genetic testing to those that are carriers of these mutations, and they will have a different type of prevention strategy. So I think this is a challenge for us to learn more about the risk factors and to design studies that allow us to develop models that predict who is at high risk and who should be targeted or prioritized for early detection.
Okay. Luis, there are several questions in the chat, so I will go one by one.
Okay.
The first is congratulations for using subtitles. It's easily to follow.
Okay, thank you.
So, congratulations for that. And there's another question about the H. pylori variation in terms of gastric cancer incidence. Are there specific strains associated with aggressiveness of gastric cancer?
Yeah, I mean, the two examples are these strains with these CAG-A and BAC-A genes. So yes, that's an excellent question. And clearly there is, I mean, there is a strong, gene environment interaction or gene bacteria, host bacteria interaction. So there have been studies in Colombia by Dr. Pelayo Correa and his group, where clearly, the interaction between the host genome or the patient genome and the bacteria genome is very important to mediate risk. But at least in Helicobacter pylori, this has been known for years now, that there are some strains that are more aggressive. And actually, my understanding is that those aggressive strains are more common in Latin America than in other, other parts of the world. So the key genes are these CAG-A and BAC-A that are the ones that characterize the aggressive disease, the aggressive strains.
Okay, there's another question about in the cases that you presented in terms of understand this tumor heterogeneity. How many of them were early stages of gastric cancer?
I think very few, because most of the patients, I mean, a good fraction of the patients that are operated at metastatic, I don't think any one of them was stage 1. Yeah, I think most of them were advanced, but that's a good question. I actually need to look at the fraction. We are actually working on the paper now.
Okay, and also in the same line, EBV positive cases among those?
There were two.
Two cases.
Yeah. One of them have a lot of mutations, and the other one didn't have any mutations. And that one, the one caveat of the data I presented is only DNA sequencing. We don't do any methylation, and we don't do any gene expression. So, for those tumors that didn't have driver mutation, it's possible they have a methylated MLH1 or methylated CDK2 and A1. It's possible that there are other things which only look at mutations. So it's possible that they have to evolve from somewhere. So cancer is a genetic disease, or a genetic or epigenetic. So they have to have genes inactivated in a way or the other. Oh, I think I lost. Okay.
Okay. There's another question about palbociclib. In the case that the resistance is used as a single agent.
Well, we are testing them individually. Okay, that is actually very preliminary. We are testing it individually, but the next step is to test the PDXs in the combination of the best chemo and the best palbociclib. And I think we are going to start with the chemos, or also to start with the chemos that are used in second line or third line therapy, because we have an eye on collaborating with clinicians to test these in patients. So we would like to work with therapies that are part of regimes of gastric cancer. But also we are seeing some of the therapies are working better. Chemotherapy that haven't been used in gastric cancer, we are seeing some interesting response. So we are trying to see what to do with that. So the goal of this study is to move to clinical trials. But I think it makes sense to start with the clinical trials that have already approved regimes for treatment, and also all the inhibitors that we are testing have been approved by have been at least past phase 1, so they are safe to use.
Okay. And an additional question is any promising biomarkers in plasma from gastric cancer for non-invasive diagnosis?
We haven't done, we are going to start doing that because we have the plasma from these patients. I suspect there will be some, but we don't have that data. Actually, Dr. Corban, I think you have something on microRNA, so we haven't done any of that yet.
Okay. But from a genetic point of view?
No, I think something that seems to be emerging in the field is that if you have a lot of ctDNA, that is another good thing. So enrichment for CT, circulating tumor DNA, tends to be not a good prognostic factor. But I haven't done much on that, so I wouldn't dare to speculate more than that.
Okay. Okay, is there any other question? At least in the chat, I don't have any more, any other questions. Feel free to open or to talk or raise your hand.
Let me see if I see the chat. Okay. Oh, I can see the chat.
It's a lot of people.
Yeah, okay.
56 people, so you have a record today.
It's good, excellent. No, actually, I enjoy working. I have a lot of colleagues and friends in Chile, so I enjoy working with you. It's a beautiful country. I went to Chile a few years ago, and I really enjoyed my time there. And then you know, as you know, we are working with a number of people, you know, in gastric cancer, in breast cancer, in bladder cancer. We have a strong links here with Chile, and so. Yeah, and this is an important disease in Chile. It's one of the, I don't know if in Colombia is the top cause of cancer mortality. I don't know how it ranks in Chile, but
in Chile it's number one.
Yeah, it is it's a bad thing to have. But hopefully, you know, we will come up with better ways to prevent it and treat it. That's the goal of our lab and the goal of many people around. So I'm hoping this is going to lead to better ways to prevent and treat the disease.
Okay. Thank you, Luis. Thanks to everybody for this wonderful lecture and all the questions and answers. That has been very educational in terms of gastric cancer.
Okay, no, thank you very much for inviting me. I enjoy my time here, and all the best for everyone out there in Chile, and take care.
Thank you. Thank you.
Okay. Bye-bye.
Bye-bye. Bye-bye.
Okay, Alejandro, I'm here.
Oh, wonderful. Okay, let me introduce Dr. Gareth Owen, who will be the chairman of the next session. So please go ahead, Gareth.
Okay, sorry. Okay, thank you. Do we have all our speakers here? Ricardo, Dr. Armisen, are you here with us?
I'm here. Hello, Gareth.
Okay, wonderful. We've run over by 45 minutes on the timing. So what I would suggest is, obviously, our two invited speakers, both Alejandro and myself, are members of ACCDiS. So our two invited speakers are yourself, Dr. Armisen, and also Dr. Lobos. So may I suggest that maybe Alejandro and myself go on standby just for now, and we start off with your talk, and then we'll discuss whether Alejandro and myself could do maybe a very quick summary of our talks, and then we give Dr. Lobos her opportunity to give her full talk as well. Okay, will that sound okay? Would you be able to speak now, Ricardo?
Sure, yeah.
Okay, I think that's the best way of doing it, because we're coming up to lunch hour. We know there's a lot of people who have to make lunch for the children, and we don't want to overrun too much. Okay,
I am one of them.
You're one of them. Wonderful. Okay, without further ado, then I just have to introduce Dr. Ricardo Armisen. He's a medical. He started off as a medical doctor, getting his medical degree from the University of Chile, and then he stayed on at the University of Chile to do his PhD. He then did a postdoc at the Howard Hughes at Stony Brook in the University of New York, and then returned to Chile to be a professor at the University of Chile. A few years ago, he left to join one of the few examples of a pharmaceutical company doing research in Chile at the Center for Excellence of Precision Medicine. And now he's left that venture, and he's now gone back to academia, where he's a professor at the Center for Genomics and Genetics at the Universidad de Desarrollo, the University of Development. Without further ado, thank you very much, Ricardo, for participating.
Thank you, Gareth. Thanks for the invitation. Can I share my screen? Let's see. Yes. Okay. A second. Let me switch. You see my full screen?
Yes, no problem.
Okay. So thanks again for the invitation. So my talk is supposed to be about functional genomics in cancer, and I think we're halfway there, as you're going to see. And we're trying to move into the functional part of the cancer genomics in Chile. Hope you will see opportunities of collaboration and joining this effort after what I showed you. So what we're about to see is a large piece of data that comes from a clinical trial called Validation of Molecular Diagnostic Technologies for Lung Cancer Patients, and its code name is Nirvana. And all of the data that you're going to see has been disclosed in several independent meetings and papers. So if you want to see more details, you can go over these disclosures. So as a quick introduction, several years ago, people on the field make some predictions about what was going to happen in cancer treatment. And one of the more influential ones from Dr. Allison, published, I believe, in Cell or something like that. He proposed that, or he realized that the cancer treatment was improving. And we have chemotherapy, we have genomically targeted therapy, as we saw in the previous talk. Then we have the immunocheckpoint therapies, and then we have combinations. And step by step, there is an improvement in the survival or the disease-free status of the lung cancer, of the overall population of cancer patients over time. And this prediction was done some years ago. And then if we start to see the data now, for example, from Germany, that they're very good keeping records of their cancer patients, something which we learned, I hope we can do here in Chile. So we can do a retrospective analysis of what happened to our patients, and they can try to replicate this prediction and into their real world data from the patients. And with a large number of them, they saw that, of course, no therapy was good, and then chemotherapy only generate an improvement. Then target therapy in patients harboring driver mutations was a good improvement too. And then immunotherapy also was a great move forward. And now probably at this time, it's going to be the time of different combinations for the treatment of advanced cancer patients, including target therapy, adjuvant therapies, and immuno-oncology. So that's a little bit of the hope that we have for the future. Then if we look at now this is a different approach. Now we're looking at the effect of cancer treatment on population mortality. And this analysis is restricted to lung cancer. You can see in this very tricky graph that if we look at the mortality adjusted by age and also by incidence, very important, you can see that the slope of these curves is changing over time. And the change that you see here in 2013 is related to the mass start of the use of this EGFR receptor therapies that are inhibitors, and they are TKI inhibitors that were approved for this. And you can see this has an effect on survival. If you look at the graph on the bottom, you can see the 2-year survival proportion moves from 26% to 35% and actually accelerates the slope of this curve. It was modified because of the introduction of the drug. This is the first evidence at the population level that these new treatments are actually improving the population mortality in lung cancer. So this is basically good news. There is something that we can do. So then, if you look today in Europe and the US, having lung cancer is of course, very tricky for the patients, but also it's very tricky for the oncologists. There is a number of biomarkers: EGFR, ALK, ROS1, BRAF, RedMed, and many others. And of course, it's immuno-oncology with tissue biomarkers like PD-L1 and complex biomarkers like tumor mutational burden and microsatellite instability that will help the selection of the best treatment in first line and second lines for these specific patients. So Chile and Latin America as a continent is probably not here yet, but this is the future that we will face in a few years. Our patients will need more biomarkers and will have more therapeutics opportunity. And of course, we need to show that there is value in these combinations of biomarkers and target therapies and immuno-oncology agents. So the results of the trial that I am going to show you was a big opportunity to take a picture of what was the status of molecular alterations in non-small cell lung cancers in Latin America. So for this study, we recruit subjects after consent in 39 hospitals. If I remember correctly, there were eight hospitals in Peru, 9 in Chile. And you can see here Arica, Coquimbo, Viña, Santiago, and then many places in Brazil, roughly, can't remember exactly, 19 different hospitals in Brazil. And we were able to invite 4,500 patients into this study. It was a very large effort that has as a sponsor were Corfo, Pfizer, and Thermo Fisher Scientific. And then for all these patients, we were able to collect very good quality data onto demographics, working habits, history of cancer, of course, biopsy, tobacco. Also, we had a liquid biopsy sub-protocol, so we collect blood samples. And specifically for the lung cancer patients, we collect results on biomarkers like ALK, EGFR, and PD-L1, among others. So, and this was done under a clinical trial, good clinical practice system. And also for all these patients, we were able to use this assay known as the OncoMind Focus assay that will allow you to get sequencing information on 52 unique genes, hotspot mutations. These are all of them are oncogenes. So these are gain of functions mutations. Also, you can look at copy number variants and fusion drivers that also are gain of functions for all these genes, doing DNA and RNA sequencing in these tumor samples. And then we had a very large challenge to classify all these mutations. So we were able to, using standard bioinformatic tools, filter out what we suppose were germline mutations, and they were removed from the analysis. And then we kept what we believe were somatic mutations, and we used several classifications to understand if these mutations were already in databases like Cosmic and OncoKB, and if these mutations were well known for clinical actionability or likely actionable or resistance mutations to therapy, or just known mutations that are recurrent in cancer. And for those mutations that were not present in the database, we used the Cancer Genome Interpreter to generate predictions about their significance, if they are actionable or they are benign or passenger. So for that, we took advantage of this engine that is available thanks to Nuria Lopez Viga's group in Barcelona, that, by the way, she will be presenting at the next meeting of the Sociedad de Genética de Chile by November 25th in a great symposium. And so you're all invited. And the Cancer Genome Interpreter is able to take novel mutations, never seen ones, and try to make a prediction if this mutation is a driver or a passenger. And they use, of course, information from many other patients, cohorts, healthy people, and they look for gene signals of positive selection, also for clustering of these mutations. Also, they take into account the gene mechanism of action, and of course, if there is evidence of mutation in the germline variant. So you get this predicted driver for these novel mutations, and the algorithm is pretty good. They have a true positive rate that is around 90% and a true negative rate that is around 85 to 80%. So the classification is expected to work. Now, if we look at the patients we recruit, they were, as I mentioned, from Brazil, Chile, Peru. We were able, into the sequencing branch of the trial, we were able to recruit more than 1,700 patients that complete the analysis. They were mostly from Chile, over 1,000, then from Brazil, and then a few from Peru. But still, it's a large number, as maybe you're not aware, but I think probably the previous speaker make notice that Hispanic population is very underrepresented in genomics, in international cancer genomics effort. So these are interesting numbers. So the age of presentation of the disease was around 65 years old. What matched what we know was the trial considered women and female and male, and also very interesting for the ethnicity, very large number of Hispanic subjects. Also, some people that consider themselves miscegenated as mixed. And so this is a very unique resource in terms of a population that actually is not part of major sequencing efforts. Most of our patients were adenocarcinoma, as expected, and for tobacco use, we have a large number of former and current, over 50% of these patients were were smokers. Had also a large number of them has a family history of cancer, around 27% of. Sorry, if we correct by the people that we don't have the data, probably close to 50% of them had an history of cancer in their families. So when you integrate all this data, you get this very complicated picture. Basically, each column is a patient and each row is a gene. You can see EGFR, KRAS, MET. They're ranked by their prevalence across. This is only the data from Chile, over 1,000 subjects. And in this particular analysis, EGFR ranks first, then KRAS, then MET, PI3 kinase, ALK, and so on and so forth. And you can see we're able to detect all kinds of genomic alterations, the different qualities of them, missense, in-frame, fusions, splice, and others. And also we collect, as I mentioned before, we collect clinical data as the biopsy stage, gender, tobacco, ethnicity, and others. So the trick now is how we make sense of all this data that has multiple layers of complexity, and let's see if we can learn a few things about them. The first things we notice is that these driver mutations in well-known cancer driver genes are well organized. You have an EGFR group of patients here, then you have KRAS, then you have MET and PI3 kinase, and this mimics what you see in the US and Europe, that usually patients will have one predominant driver genes in their tumor. So we're not different, but also we have patients with multiple mutations that are very interesting to take a deeper look into them. Then, well, you can see here where the patients that actually finish the sequencing are from, most of them from Lima, Santiago, a bit from Concepcion, and then in Brazil, many of them from Sao Paulo, but also from other cities. I'm not sure if I mentioned it, but if you look at the pathological stage at the time of the patient entered the study, and actually for many of them was at the time of the original diagnosis of cancer. Regrettably, many of them are stage 4. In Chile, large, I believe, 70–80% of them. In Brazil, slightly better, 60–55%. And in Peru, well, if you have to correct for the known, that probably many of the unknown are actually advanced tumors. Probably also is over 50–60% of the patients being diagnosed at a later stage. And now if you look at the genes, and we use the clinical actionability categories, you can see the EGFR mutations are actionable in a very large percentage, as expected. Then you have KRAS with many known mutations that are well described in other tumor types, but are not directly actionable in lung cancer. And then you start to see this green here that are novel mutations, and this get our attention because you can see many genes that are actually very interesting and very actionable. You can see there is some a very high proportion of novel mutations in some of them, and a very long tails of genes that are not very frequent in the whole cohort, but many of them are actually targetable, like ROS1 and others. Also, for example, ERBB2, or most known as HER2. So then if we look at this busy table here, you can see that for specific mutations, for example, in the MET gene, we're going to put a lot of attention in the MET gene. This is a well-described actionable mutation in many cohorts, including lung cancer. It's the skipping of exon 14. And we got a large number of subjects, and very interestingly, this is enriched in Chile. Chile is very particular about these mutations, has a large proportion of patients with these particular mutations compared to Brazil and Peru. And the same happened, these are specific EML4 ALK gene fusions, for example, in the ALK gene that drives our expression and gain of function of the ALK genes. Also, you can see that this enriched in, for example, in Brazil compared to other countries. And the same apply for mutations in red, ROS1, well, EGFR, very well-known gene, very important in lung cancer. Also, there is some regional distribution that we will, in further studies, we expect to see if related to ancestry or not. That also, we have some data on exposure to external factors that may generate a different pattern of mutations. That are research questions that we're still considering. And for novel variants, we got some good numbers in the ALK gene. specifically in the MET genes. Also, we have some variants that were never seen before in other countries or in other cancers cohort, also for red and ROS1. And these novel variants are very challenging because right now this means that these patients, well, we don't know if we believe, because we use the cancer genome interpreter, that these novel variants could be drivers of the cancer in those patients, but actually nobody knows if these mutations will respond to the target therapy that was approved for ALK, MET, RED, and ROS1 in the very same patient population. So at this point, these patients will not benefit from the knowledge of this mutational pattern on their genes. So now if we move to a more friendly approach, you can see for very important genes like PI3 kinase, MET, KRAS, EGFR, and ALK, we were able to find a large number of likely actionable mutations, but also there is this proportion of novel here that we really want to focus our efforts on, because for all these actionable and likely actionable, the problem for the patients to get the drug is not a scientific problem, it's an access problem. It's having the right insurance, having being the drug available in the country and considerations like that. But for this population here is different. We don't know if they will benefit from the drug. And if we look at the clinical characteristics of these patients, they're very, very interesting. And they're not if we look at the family history of cancer, the proportion of females, age, and a parameter that characterize the mutations, we will see there are different distributions suggesting different biologies that we need to look deeper. For gene fusions, you can see here ALK is fused mostly with ENL4 as expected, also RED with KF5B, and also for MET, a very large number of intragene fusions between exon 13 and 15. These are all well-known driver gene fusions that are important in lung cancer. So finally, if we go to the protein where the action is supposed to happen, and we put these novel mutations in context, for example, for the MET gene, you can see here, these are the different domains of the protein. You have the tyrosine kinase domains, you have regulatory and signaling domains. Many of the novel mutations are actually in the juxtamembrane domain, and also close to this tyrosine kinase domain that MET, also the same for RED. Most of the novel mutations are here in the tyrosine kinase domain, also for ROS1, and also for ALK and for ERBB2. So we're really looking forward to realize if these novel mutations are actually driving the tumorigenic process of NOT. Just as an example, not so long ago, FDA approved this particular drug called capmatinib for the patients that have this particular exon 14 skipping mutations in the gene MET, and also MET amplifications. And you can see in these plots that the patients that were previously treated or previously untreated, they have a large number of them has a partial response to this new drug, and this granted the approval for further study, clinical trials, and use in very specific patients. But right now, the patients that we have with MET novel mutations are not going to benefit from this drug, and they're not going to be part of clinical trials, not only because they're here in Latin America, but also because nobody's looking for these mutations when they want to actually do the assay. So if we go back to the MET gene in our population, here you have the mutations. Our patients, they do have the MET exon 14 skipping mutation, but also they carry, we select these two mutations for further study. You're going to see this is a work with Solange, a new postdoc in the lab that is going to start working next year. So here is the actionability of the MET gene. We got this yesterday from the database of actionable genes. You can see the mutations that are linked to the therapy, and these are not part of the label of this drug, not only in non-small cell lung cancer, but also in other tumors have not been described yet. So we believe it's important to do the effort to understand the role of these new mutations that actually present only in patients from Latin America so far. So where is the functional part of this? Well, it's mostly an invitation. We have two new projects that we hope, well, this one got funded recently, and we hope to work with Dr. Owen, Garrido, Marceline, Corbalan, and Ibanez in the discovery of novel actionable genomic alterations in South American cancer patients using the data that I already showed you, and also data from gastric cancer, breast, colon, and gallbladder cancer that we already have over 2,500 patients that were already sequenced by this group, and we expect to be able to validate and segregate a novel set of variants, be very aggressive to understand if there are drivers or not. Then, of course, there is a challenge that I didn't mention, but as you may be aware, the reference genome that we call humans is actually very depleted of people from Chile, Peru, and Brazil. We're not part of this big effort. That's a technical problem that we need to address. Well, and then we aspire eventually to validate these novel mutations through in vitro and in vivo analysis. And that's the second project with Solange Rivas, that she took the challenge of these novel mutations in the MET gene, these particular two ones, and she expects to see if they're able to confer transformation capacity when you express them in normal cells, like lung cancer cells, lung normal cells. Also see if these mutations now in a tumor background drive sensibility or not to specific target therapies, not only capamitinib, but there are many others that are new therapies for this particular MET gain of function. And then finally, of course, if we have enough time and energy, I hope we can study this in a xenograft in vivo model of non-small cell lung cancer, see if in an actual mice, these mutations drive tumorigenesis, tumor growth, and progression. So this is the functional part of our work that we expect to carry in the near-term future. Finally, I would like to thank you a large number of people. You can see here all the investigators from Brazil, particularly Elano Freitas from ACCDiS Camargo Center that plays the role of the coordinators in Brazil. And then in Chile, we had a very large contribution for Dr. Chernilo, Dr. Karen, and from Instituto Nacional del Tórax and Research Cancers Associated Research Cancer Center, also from Peru, that we were able to collect very good samples. And of course, very important, all the people that did the sequencing in 4 central labs that did all the actually sequencing work and part of the pathology work at Universidad Mayor and ACCDiS Camargo Cancer Center in Sao Paulo, Brazil, and Catherine Marcelin in the Laboratorio de Genómica del Cáncer at Universidad de Chile. Also thanks to Cristina Fernandez that did most of the pathology work. Thank you very much for the time.
Okay, thank you very much, Ricardo. Excellent talk, and thank you for being exactly 25 minutes, right on the second. Wonderful. So as we mentioned, as in the first session, we're going to have questions at the very end of this block, which is, I've got my questions written down. If you have any more questions, please put them in the chat. And I've just been discussing offline with Dr. Corbalan. What we're going to do is Dr. Corbalan and myself now are going to present for 10 minutes each, no more than that. And then we're going to have the full talk of Dr. Lorena Lobos at 12:30. Okay, and then we still have another around 5 minutes for discussion at the end to finish on time. Okay, so this is being a challenge now that we have to, if you could stop sharing screen,
sure.
Ricardo, and I will, Alejandro, would you like to go first or will I go first?
Whatever you want.
Okay, I'll go first then. If you could let me share screen, please.
Okay.
Oh yeah, okay. Okay, can you see my screen?
Yes.
Yes, all right, wonderful. I'll turn the microphone off. My Wi-Fi is fine, but I'm pretty ugly, so it's better if I just mute this straight away. Okay, so in 10 minutes, this is what I've got. I've cut my chalk down to. So what I'm going to tell you now is a story about how we tried to use clinical data that we collected as in a gastric cancer task force here in Chile, together with many of the actors that Ricardo just mentioned. And the idea was to predict chemotherapy-related toxicity in Chilean gastric cancer patients. So there are many factors that determine how we respond to drugs. One of them is sex. Women seem to have more toxicity than men. Age, our physical health, the renal hepatic function, drug-drug interactions, of what sort of therapy we're actually receiving, and of course, genetic factors. In fact, there's several papers now stating that 20 to 40% of this interpersonal variability to drugs comes from a genetic factor. So our question was quite simple. We're going to use small nucleotide polymorphisms, which are just changes in the DNA sequence through populations. For example, Dr. Corvalán in the same gene could have an A, I could have a G, and Dr. Armisen could have a T. Okay, neither one of us is a mutant, to my knowledge. It's just the natural distribution of genetic sequence within a population. But we were thinking, well, what happens if some of these, if this was the promoter of an enzyme that metabolizes drugs, and maybe in one genome, this A here would mean a transcription factor would bind with more or less affinity, which means there'd be more or less enzyme that we'd be metabolizing, and therefore there could be more or less incidence of toxicity. So that was our general idea. And based on this, we decided to see if we can do the background work to one day develop a test. And I'm going to explain what a nomogram is in a little bit, to combining several SNPs that we identified with clinical data to predict toxicity to generally FOLFOX and FOLFIRI, which are fluoropyrimidine and platinum chemotherapies currently used for treating gastric cancer. So cutting a very long story short, what we have here, we're collecting Chilean patients. We have, thanks to our medical oncologist, Dr. Marcelo Garrido, we have a very good database of the patients and their clinical data. We decide to bring them into our study, and then we look at several SNPs, genetic sequences, and all the clinical data. How did we determine these genetic sequences to use? Well, we looked in our data in the Pharma GK database, okay, and PubMed. We looked at what was present within the South American population, because it's great to have a SNP that's very strongly associated with toxicity to platinum drugs. But if it's not present in the South American population, it's not really worth looking. And also we looked at the literature, okay? So what we did is we put all these results into a big algorithm that my doctoral student, Miguel Córdoba, developed. And then we came up with a selection of 12 SNPs that we were going to look at. We then started to put all the data together. We looked at some classification algorithms, and we did a training set and a test set. Then we came out with a classification algorithm that gave us a ROC value. And then we started putting this into a clinical applicability model. It was called a nomogram, where we could actually maybe predict whether a patient would or would not respond. Now, this sounds very simple on paper. We just put all the information into a classification and algorithm. But really, this technology wasn't in my laboratory. I don't think it was actually even in Chile when we started this project. So what we did is we looked outside of Chile, and we found a collaborator at the Johns Hopkins University in Johns Hopkins Hospital in Baltimore in the United States, and a collaborator called a doctor called Li Jiwang. Now, just at that time, Li Jiwang was actually returning from the Johns Hopkins to start his own laboratory in Dalian, and Dalian is in China, and it's just it's about 400 kilometers from the border of North Korea. So he was moving back there. So what would be perfect for us if I could send a doctoral student to China to actually participate in and learn how to do all these bioinformatics, all these algorithms that we needed for this project. So we decided to send our doctoral student, Miguel Córdoba, and he visited for 11 months. And I'd just like to say personally now, thank you to the Chinese embassy for giving us this grant. They paid for his travel, and they paid for his his time for actually living in China for 11 months. Now, Miguel, I have to be honest with you, was not that keen to go to China, especially when we told him that we had to go to the border of North Korea. And at that time, North Korea were firing long-range ballistic missiles over the Japanese Sea. So he wasn't that keen, but we packed his bags for him. We pushed him on a plane, and he was very happy. So he was actually visited by some very famous people within his first week when he was in China. Actually, Kim Jong-un actually decided to have a meeting with the Chinese president, Xi Jinping, in Dalian University. So Miguel got to be there with two great leaders. And his apprehension slowly went away. And 11 months later, when he came back, he absolutely loved China, and he wants to go back now to do his postdoc. He just graduated from the University of Chile a few weeks ago, and he wants to go back maybe to South Korea or to China to do his postdoc. Okay. Anyway, let's back to the science. So what Miguel was doing, he was learning how to do classification algorithms such as logistic regression, super vector machines, decision trees, random forests. And we came up, cutting this to a very, very short thing, we came up with one model which had 4 or 5 items of clinical data, along with 3 independent small single nucleotide polymorphisms. And when we looked at that, we actually had one combination that gave us very high sensitivity, very good specificity, and very good accuracy. Sensitivity means we got a lot of positive, what we thought would be toxicity were in fact toxicity. When we predicted that there wouldn't be toxicity, we in fact didn't get toxicity. And this accuracy is sort of a little algorithm that combines both of these values to have a good score. And we got an area under the curve of 0.82, which is very respectable indeed for actually going to the clinic in the future and predicting and using this to help guide patients. So what we then did is we then used this this algorithm, and we went back and analyzed every single patient that were in this study as a case control study. And what we then is we designated points to the age, because the age does matter. We found out that there is statistical correlation to sex. Women do seem to have statistically more prevalence of toxicity, the chemotherapy regime, what SNPs were actually present. And then we got, at the end of this, a total score, okay, which then converted into a probability. So what we did then is then plotted our test score value against whether our patients, because this is a retrospective study, that didn't have toxicity and patients that did have toxicity. And as you see, we get a nice distribution here of patients that have a high score value when they have toxicity, and they have a low score value, even more so, when there is when there's no toxicity. So what we see here is we have a high score, which we could say we have to now go to the next validation stage of this assay, which we're trying to set up at the Catholic University, where we have these patients here have a high probability of toxicity, and these patients here will have a low probability of toxicity. So what we could, what we found now is this is more than just looking at patients who are going to receive FOLFOX or FOLFIRI or any chemotherapy based in 5-FU and platinum drugs, because there's now some studies that came out very recently, where we're combining chemotherapy with immunotherapy. And interestingly, the last study that came out showed that the chemotherapy, when combined with immunotherapy in gastric cancer patients, actually give better results for the patients. But one of the problems was that many of these patients getting this combined chemotherapy and immunotherapy had to drop out of the study because the combination of chemotherapy and immunotherapy, it was too toxic. Okay, so what I'm thinking now is maybe to sell this to the pharmaceutical company or to bring it into our next validation trial, is that maybe these patients here that we identify with a high score with our test, these patients here could go just to immunotherapy as a monotherapy, But these patients here who we predict will have very low chance of having toxicity with chemotherapy. These will be excellent, and it's actually the majority of patients. This could be excellent candidates to receive the combination of chemotherapy and immunotherapy. Okay, so there we go. I'd just like to say thank you to my collaborators. This could not have been done without the work of Miguel Corvada, who went to China, and also my collaborator in China. Thank you for the guys at, I didn't mention it because I had to cut all their talk all their results out of the talk today, but thank you to Semp, who really helped us. Thank you to Dr. Quiñones, who was also co-tutor to Miguel Corvada, and also to the medical team at the Catholic University, to Dr. Marcelo Garrido, Ignacio, Mauricio, Loreto, really for helping me, helping us, because we work together, getting all these patients together. Okay, so thank you for that. And with that, I think we've had quite enough of me, and we'll now move on to a talk by Dr. Alejandro Corvalán, who will talk for 10 minutes, just like me, on his results. Okay, Alejandro, all yours. Actually, no, sorry, I have to introduce you. I forgot. Sorry. All of you know Dr. Alejandro Corvalán, certainly listening to this talk. He's also a medical doctor and pathologist by trade. And he did his postgraduate studies at the MD Anderson, and he also spent some time in Japan before coming back and taking up as a professor and investigator in cancer at the Institute, at the School of Medicine at the Catholic University in Chile. And he's also a director, one of the 6 directors of the ACCDiS Institute. Dr. Corvalán, all yours.
Okay. Have you seen the screen?
Yes, perfectly.
Okay. Okay, so thanks, Gareth. I will going to talk about the epigenomic aspect of mostly gastric cancer. I try gastrointestinal, but will be gastric cancer in the end. Well, gastric cancer, as you know, has 2 major pathways. One is the environmental pathway, and the other one is genetic, host genetic factors. And I will focus on this pathway, which is mostly associated with the diffuse type of gastric cancer. This diffuse type gastric cancer is characterized by the inactivation of a single gene, which is HICADIR, which is a cell adhesion gene. And this is immunohistochemistry showing normal expression of HICADIR. And in diffuse type gastric cancers, it's totally lost expression by several mechanisms that I'm going to address now. And one of them is the familial type of gastric cancer, which is, as Dr. Carvajal mentioned, is diffuse gastric cancer, which could be run in families like this family, kindred, or could be an individual cases in which is only a single case, young age, with no family history. This is one of the important aspects, and usually less than 50 years old. So it could be family history is important, but it's not totally necessary. And the second important aspect that is associated with the breast cancer family. So hereditary gastric cancer is not an isolated disease run with breast cancer too. So in both ways are quite related for unknown reasons. So with that, we start a collaboration with Dr. Enrique Norero, who's a surgeon from Hospital Sodero Rio. And he established a cohort of 36 patients that were tested for CDH1 mutation, and to identify only 1 case with pathogenic mutation. All of the rest were non-coding located or benign, which represent 2.7%. And this is the case of the family, in which only there's 1 case, young age, 22 years old, and no family history. But we tested the whole family, and all these dark circles here means that they were mutated. So one of the carriers were the sister, which was, she accepted surgery, and this is the prophylactic gastrectomy. This is the stomach of the patient who was healthy but carried the mutation. And we identified diffuse gastric cancer, which I'm showing here, in this tissue. So this was the first case of prophylactic gastrectomy in the country, in Chile. This has public health impact because after this finding, hereditary tumor group was established at Hospital Sotero del Rio, which is the first hereditary group that has genetic testing using different platforms. Also, there is a breast cancer surgeon involved in the group, and 2 nurses, one focusing on tumor register and the other one in genetic counseling, and is in the public system. So it's something that has a real impact in terms of public health. But from a research point of view, this was a little bit disappointed because we were expecting up to 19% of frequency of CDH1 mutations. So we established another collaboration, one with Dr. Luis Carvajal. As he already mentioned, we collaborate in the identification of DNA recombination repair genes, which represent 6.5% of hereditary diffuse gastric cancer. And more recently, Graciela Molina, who's a postdoc working with Luis Carvajal, identified another mutation in a gene that's called CTNNA1, which is part of a CDH1 complex. So with that, we are expanding the role of different coding genes associated with hereditary diffuse gastric cancer. But we're interested in epigenetic landscape, which means that what's going on outside of this. So we can focus on non-coding RNA that mostly target 5 untranslated regions, or microRNA that target and inactivate coding genes by binding the 3 untranslated regions. So we focus in microRNA, and that was based on this publication by Lilian Jara group. Thomas Mayo is the leader of this author of this publication. And he identified in a family that is a breast and gastric cancer. These are the breast cases, breast cancer cases, and gastric cancer case, in which there's a deletion in the germline of the microRNA cluster 1722, which are 6 microRNAs, and the deletion was present here between microRNA 17 and 18A, and this deletion was a 6 base pair deletion, which predict change of the structure, and not the same group, but a similar, another independent group showed that deletion in this region was associated with an increase of expression. So based on that, we tested what the possibility that this cluster was associated with an activation, epigenetic activation of CDH1 gene, and this was done by Natalia Landeros. That she identified that by mid-walk analysis, all these cluster, all the microRNA from this cluster were able to bind the 3 untranslated region of CDH1. And in the vivo analysis showed that in TCGA, the expression was inverse correlated in tumor, but not in normal adjacent tumor. And one by one, MIR18 was significantly inverse correlated with the expression of CDH1 of Hikadir. So based on that, she developed an assay for tested a large cohort of families looking for this deletion. And this is an example. This is a homoduplex where there's no deletion. And when it's a deletion, a heteroduplex is found between the wild type and the 6 base pair deleted sequence. And after testing 100 families, no, we didn't identify any deletion. So this also was a little bit disappointing, but we're still looking for other microRNA that would be associated. And also, we think that this cluster is quite important in cancer, so probably we will test the whole cluster in these families. The other aspect of epigenetic landscape of gastric cancers is the Epstein-Barr. EBV was identified by this group of 4 people back in 1964, and is a 200,000 base pair DNA virus that's seen as an episome. And in 1992, Shibata and Weiss found that the virus is present in gastric cancer, which was not well accepted until 2014, when EBV was included in the molecular classification of gastric cancer. So take almost 30 years, 20 years almost to accept this association, which is a second etiological agent for gastric cancer. In between, there are several groups, including myself, that were working in the association of EBV gastric cancer. And this was consolidated by Constanza Camargo, who created the NCI EBV Gastric Cancer Consortium. So we put all the cases together, and for our surprise, Chile is probably the top one in terms of incidence and prevalence of this association, with a lot of variation around the world, but they tend to be in the Americas, high frequency. This data was validated by the group of Gareth Owen. Miguel Cordova identified 13% last year, so quite similar frequency. So this was validated. With that, we put a proposal led by Keila Torres about EBV-associated gastric cancer based on microRNA that has been recently described in this tumor. And I will move fast. Basically, what we found that these are target genes that bind this microRNA, but also non-coding RNA. So there's a kind of balance. And basically, what we think that this could be explained between this balance between the sponge by non-coding RNA and messenger RNA. This could be an explanation why we have we acquire infection early in life, but rarely we develop disease, and probably because these microRNA are sponged by non-coding RNA. But with age, these non-coding RNA disappear, and they are able to identify to target apoptosis-related genes, and apoptosis is the loss of apoptosis is the cause associated with development of gastric cancer. This is a seed match between a specific non-coding RNA and BART, and also we have preliminary data showing that these cases are EBV positive here. They lost apoptosis in the EBV positive cases, and this non-coding RNA is lost in EBV-associated gastric cancer. So, and finally, to combine both approaches, we identified that there's some previous publication that found that there's some mimic or very similar sequence between microRNA from the virus and microRNA from the host. So in that way, we can combine the EBV-associated gastric cancer and the hereditary diffuse gastric cancer. And I'm showing here, this is a pilot study in which there's a loss of expression of kikadirin and increased expression of MIR18 and MIR microRNA from the virus. So in that way, we can unify or combine two different aspects by epigenetics in the diffuse type of gastric cancer, the hereditary point of view of gastric cancer and the EBV-associated aspect by this microRNA. So this is my group, and I want to take a moment to mention that Eva Polagoikova is not with us since 2 weeks ago, and she was a key person to help Kayla and Natalia to produce the data that I have been showing. And these are the grants that support these results. Thank you very much.
Thank you very much, Alejandro. Wonderful to see another photograph of Eva. Now we'll move on to our final talk, which will be given by Dr. Lorena Lobos. Dr. Lorena Lobos is a biochemist from the University of Valparaiso, and she did her master's degree and her PhD at the University of Chile. And now she's entering the faculty track at the Center for Regenerative Medicine within the Faculty of Medicine at the University at Universidad de Desarrollo here in Santiago in Chile. Lorena, thank you very much for joining us today, and the screen is all yours.
Thank you, Gareth. I have returned.
Okay, all looks good. We can see your slides.
You listen to me well?
All's good. Carry on. Yes.
Wait, I have a problem with the... I can't listen to you again.
If you can hear me, we can hear you fine.
Okay. Today, I will present you the research that I have led almost 3 years. First, like an introduction, I want to tell you the impressive increase of the number of publications relative to the cellular communication in cancer, specifically in the exosome communication. Consider the difficult charge cargo of the exosome. So in this challenge, our research group also joined in breast cancer. The most common to this publication is the study how the exosome are capacity to promote tumorigenic or metastatic conditions, and begin so continuously this idea, we try to evaluate how exosomes secreted by metastatic cells promote invasion and metastatic property like tumoroid formation. In this slide, you can see here 2 recipient cells like a T47G and MCF7 that are tumorigenic cells, and in presence of the exosomes to the metastatic cells, the capacity of migration and invasion increase almost 5 times. And here you can see how the possibility to form tumoroid formation increase too in presence of the same exosomes. So what is the role to do this exosome? What is the cargo of the exosome to contribute to increase these tumorigenic properties? In this line, our group of investigation beginning to try to understand what is the charge of proteomic in the metastatic exosome. For evaluate that, we evaluate the proteomic analysis of the exosomes, and we obtain in the biological process in the category, and the cellular process are increases, like a cellular component organization, cellular metabolic process, or response to stimulants, and increase on signal introduction. in this evaluation, we obtain several proteins involved in epithelial mesenchymal transition and involved in metastatic promotion. But one of these proteins in the cargo of the exosome take our attention. This is lactaderine. The importance of this protein is because this protein in normal and physiologic process is participate in lactancy property. This is a protein to maintain the contractile units in the lactancy property, increase angiogenesis, and is acting like immunomodulator in this process. But when this protein is present in pathologic process, like a breast cancer or another disease, this protein is promoter of epithelial mesenchymal transition, invasion, migration, and is described like metastasis promoter. If we review the history of the publication to relate it with lactaderine, we can build this time course of the publication of lactaderine, and in of the time into 1808 until now, lactaderine has been described as a marker of tumorigenicity in solid tumor tissue, not only in breast cancer. You can see here in 2000, lactaderine preclinical models in melanoma or colon, two are described like a tumor promoter. If you see in detail this time course, have a jump between the publication. So the most important publication in the involved to this described like possible biomarkers is until 2018, when lactaderine in solid tumor is described like increased metastasis in lymph node in patients with breast cancer. And in 2002, we published the described the lactaderine is present in extracellular vesicles. So taking this idea to lactaderine is described like a marker of tumor genicity in solid tumor, we want to build and reconstruct to these jumps to slides. For that, the first was described lactaderine in how is the distribution in different cells of breast cancer. If you can see here in normal epithelial cells and 3 different tumorigenic cells line and a triple negative breast cancer cell line, here a metastatic potential. And you can see here in green the detection of lactaderine in all cells in breast cancer, and in normal epithelial cell, the levels are very lower. Continue with this characterization, lactaderine expression was detected increases in all tumorigenic and metastatic cells, you can see here, and compare with the epithelial cells. And if we detect the protein in white Western blot, you can see here a representative example. Oh, sorry. You can see here the level of the protein are detected in higher in the tumorigenic cell line compared with the metastatic cell, but in all cases is more compared with the epithelial cells. Continue with this idea and take the biodistribution or the distribution of the cells on the lactaderine in the cells. We try to evaluate if lactaderine is present in different compartmental cells, like a superficie of the cell surface compared with the cytoplasma in the cells. By cytometry, we evaluate the cell surface of the presence of lactaderine. In the case of the metastatic cells, you can see here more detection compared with the tumorigenic cells. But how is the cellular distribution now if this same cell formed than tumor in mammalian gland, and how is the lactaderine is present or not in plasmatic exosome in this might to form this tumor formation. For that, we first to standardize the orthotopic model of breast cancer cell line, and you can see here when we injected the tumoral cells in the mammary gland, we rate and evaluate the tumor formation in the time, and when the tumor volume are rated to 100 cubic millimeters, we do the euthanasia and evaluate the tumor tissue. If for a representative example here, you can show the tumor tissue and the loss of the architecture of the mammary gland, and with this model standardized, we evaluate the level of lactaderine in different tumor formated with the different cell type. Here is a hematoxylation, and in green again, like by immunofluorescence, the detection of lactaderine. So we can evaluate and detect the lactaderine in cells and the tumor formed in mammary gland in orthotopic models. So lactaderine is present in plasmatic exosome or not in this mice model. For that, when we do the euthanasia, we take at the same time blood samples, and thinking in the positive control, we take blood of the mice in lactancy time, and like a negative control, we take a blood of the younger mice. Is like an example here, a representative Western blot. You can see here how the positive control is by Western blot detected lactaderine. In young mice, we don't detect the protein present in the exosomes. And in mice with the tumor formated rated to 100 cubic millimeters, we detect the lactaderine in the exosome. TCG1 is a positive control for exosomes, and calnexin is negative control for reticulum endoplasmic contamination. So lactaderine is present in plasmatic exosomes secreted by the tumor formated in mammary gland in the Murino model. But the next, or the question we do is, what are the capacities that could be being promoted by lactaderine present in these exosomes? We continue to evaluate, so we exosomes secreted by different cell lines are purified and characterized. We show you here the distribution is the range we expected. In the integrity, you can see here in the microscopy, electronic microscopy is okay. So if a question important is if we purify exosome by ultracentrifugation is a classic method, or using a exospin column system, we evaluate and we detect in all conditions of different purification lactaderine in exosome compared with the cells. So again, positive controls like TCG1 or Alex, or control negative like contamination and GM130 or calnexins. So lactaderine is present in exosome in plasmatic mice model, is present in solid tumor. So what is a possible role in invasion in capacity in vitro before to try to understand what happened in vivo? So for that, we're beginning with the classic assays, and thinking in the translational study, we not only to interference the expression in the cell of the lactaderine. We try to use the idea to the immunotherapy. So we try to block the lactaderine present in the exosomes. So if you see here, when the recipient cell is a MCF7 cell, is a tumorigenic cells, these cells, when receive exosome blocked with lactoferrin here, this cell decrease the invasive capacity compared when the cells receive the exosome not blocked. And if you put the antibody alone, the effect is almost the same. This effect is when the cellular recipient is less aggressive compared with the exosome to secrete it. It's a paracrine effect. If you evaluate the autocrine effect when the cell recipient is the same cell to secrete the exosomes, the effect is the same. When you block the exosome with antibody anti-lactoferrin, or if you put only the antibody alone, the capacity to invasion is decreases. So the block of lactoferrin with anti-lactoferrin decrease the invasion capacity in this assays. Well, thinking in what is the most difficult capacity to in vivo development the metastatic state in the patient, we try to evaluate the capacity of grow independent in the cells. So for that, we evaluate in clonogenic site using the recipient cells, MCF7 cells, and using the same strategy to block the lactoferrin exosomes with antibody. You can see here how the clonogenic capacity is decreased when we use the antibody alone or the antibody blocked the lactoferrin in the exosomes. So this happen in vivo, have some effect if we do in vivo assay. For this, we prepare a specific strategy. It's a peritoneal breast carcinomatosis model, and this model is very similar to the late stage in the patient when the woman have ACCDiS malignant in the late stage. So the cells are injected in intraperitoneal cavity of the mice, and after that we administrate to mice different exosome or the exosome blocked with anti-lactoferrin or antibody to anti-lactoferrin alone. So when the carcinomatosis model is beginning to growing in the peritoneal cavity, the mice beginning to observe an increase on the abdominal volume. So it's very fast to show at the 21 day how this phenotype in the body of the mice is changed. The results of the experiment are very difficult to understand, but some happen very important to happen. And it is a result of the ACCDiS malignant. If you see here, when mice receive the administration of the antibody alone, none of the mice present malignant ACCDiS. And when the mice receive two exosome blocked with antibody with anti-lactoferrin, only one of them developed ACCDiS malignant. I see here the distribution of the mice and the formation of cell ACCDiS. So if you compare the mice who not receive any exosomes or any treatment, don't form ACCDiS malignant. When you inject the exosomes to mice, these mice increase the metastasis behavior, and the blockage alone or exosome with blocked with antibody, this behavior is decreased. So lactoferrin will be a good therapeutic target or diagnostic in breast cancer patient. I want to show you, this is a very preliminary results. I need to work much in this idea, but we work with Thomas Laboratorio, and this laboratory is in Coyhaique. They have 75 women with breast cancer. Some of these women try to understand what happened with this evolution, and this is a question to all patients do it when have a breast cancer or any cancer. So some of these women, they give us a sample of blood. With this sample of blood with purified exosomes, you can see here the nanotracking analysis for evaluate the quality of the purification, and the diameter of this exosome are in the mean of the we expected. And after that, we beginning to standardize the Western blot for detected lactoferrin in the exosome of patient. We use like a negative control, a woman with a recently exam of mammography with any detection, and woman have 23 years after using the blood sample of the woman with breast milk and in lactancy period. And after that, we have show you here 5 samples of exosomes of a woman with different state of the breast cancer. Stage 2 of in situ carcinoma, or stage 2 with post-mastectomy recently, or stage 3 in treatment with tamoxifen, and inclusive here, the most higher detection in this Western blot is the detection is the lactoferrin and exosome of a woman with a recently metastasis in ovary. And this Western blot is not very, very good because it's the band is very diffuse. So, but that we use immunoprecipitation for lactoferrin for try to understand if it's okay, is a specific this detection. And you can see here, we detected lactoferrin in the same condition, and when we don't use the antibody, we don't detect nothing, is talk and suggest the specific detection. So lactoferrin is present in plasmatic exosome of breast cancer patient, and maybe is a tumorigenic biomarker. In conclusion, at least we can say lactoferrin is present into exosome of a metastatic cell line, and this present is contribute to increase the invasion capability and malignancy and carcinomatosis, breast cancer model, and the immunoblock of lactoferrin in exosome maybe decrease this property. The next step is try to evaluate if it really lactoferrin in plasmatic exosome is or not a tumorigenic or metastatic biomarker. Finally, I want to say thank you to our laboratory, and specifically to Eduardo, Matias, Valentina. He worked a lot in our investigation. Thank you very much.
Okay, Lorena, thank you very much indeed. Very, very nice presentation. Okay, could we all join in? Ricardo, could you, Alejandro, could you put your camera on? Okay, we're in the hour, so let's just do quick, maybe just 10 minutes and just finish 10 minutes late. A few questions that have come up. Lorena, are you there?
Yeah.
All right, we'll start straight off with a question for you. A question about lactoferrin coming from Dr. Sanchez in Valdivia. She's asking, does lactoferrin promote binding to the endothelial cells? If so, what receptors is it using?
Lactoferrin have two domains. One of these have a RGD domain and interacted with integrin alpha B beta 3. And for the other side, have a EGF-like domain and interacting with the fosfatidylserine in this other side of the structure of the protein. So a specific receptor is not described, and so it's very important to interaction with this integrin alpha B beta 3 or alpha B beta 5.
Okay. All right, thank you. Dr. Armerson, we have a question for you regarding the MET exon 14 skipping that's highly prevalent in Chilean patients. What is known about the functional consequence of this isoform?
Sure. So that's a gain of function modification. So the skip exon will generate a functional protein, but will have a larger tyrosine kinase activity driving signaling pathways that are underneath that receptor. So yes, a gain of function, clearly.
I actually have a question on your percentages of mutations. I published a paper a while ago with a Chinese group on lung cancer. And what we found is that EGFR was a lot higher, was the predominant mutation in lung cancer in China, as opposed to KRAS being the predominant more frequent mutation in European and American populations or North American populations. But I noticed from your results that you also have a very high EGFR mutation rate. So that, have you noticed any difference between your statistical mutation rate and the other genomes?
So yeah, so EGFR mutation rate is larger in Peru, and and then in Brazil, and then Chile, and the underlying theory and underneath that is the influence of Asia populations. And so Sao Paulo has a very large Japanese population, and in Peru you also have a very large component from Japan. So it seems that the EGFR mutations came with the ancestry, with the Asian ancestry. That's one theory, at least so far. Yeah, we saw that, and also you have a switch in the rate of KRAS to EGFR. So it makes sense when you say, yeah.
Yeah, that makes sense. Yes, it's the Chinese ancestry and Asian ancestry in Peru. A question for Dr. Lorena Lobos. Lactoferrin is an outer protein in the membrane of the exome. I was wondering, I'd like to combine a question for you as well. Could you also, when you're answering, I was wondering if lactoferrin is an estrogen-regulated gene. Obviously, it's a pregnancy-related gene. So is it estrogen-regulated? So I was just wondering, maybe in the future, in breast cancer positive patients, could the presence of tamoxifen, which is obviously an estrogen receptor inhibitor, could that be reducing the amount of lactoferrin in exosomes, and then maybe reducing the amount of metastasis? Because it's a very good drug that works very well.
Okay, it's very nice question. First, I don't show you today, but we use the strategy to take or to use the proteinase K for incubate the exosomes. So when incubate the exosome with proteinase K, we lost the function, the change in the functional event in the increase of the migration with the exosome. So for this idea, we try to understand the lactoferrin is this transmembrane protein is outside in the exosomes. And when you use, it's the reason because try to use the block of the lactoferrin with antibodies. I don't listen.
Sorry. We have another question here. Solange is asking, does immunotherapy cause autoimmune disease? And the answer is yes, it is actually one of the side effects of immunotherapy. Even a few of the patients actually at the Catholic University where we've seen where they have a very positive response to immunotherapy, and stage 4 cancer has actually regressed, gastric cancer. These patients actually were left with diabetes type 1, which is an autoimmune disease. So it is a problem. It is one of the side effects of this cancer treatment. So just to finish up, I would like to one last question for Dr. Armisen is, I'm going to read it straight away. In the future study focused on the discovery of new actionable mutations in South America, do you know how, what is the proportion of cancer patients you'll be looking at in terms of cancer population of each country and types of cancer?
So right now we have data from Chile a lot, and then from Brazil and Peru. In terms of the type of cancer, all the data that we have in Peru and Brazil is from lung cancer, and we are going to develop a strategy to look for collaborations. We expect to get really good at doing the analysis of the data. So people from Peru and people from Brazil, we hope will want to collaborate with us and contribute with data that they may have in other cancer types. We're aware of a large gallbladder cancer project running in Latin America by Dr. Marceline. And also there are several efforts in gastric cancer in Brazil and also in Peru that we hope we can invite them to join the study. In Chile, we are lucky. We have breast cancer, we have gastric, colorectal, and gallbladder and lung cancer. So it's a much more comprehensive analysis that we can do here in Chile, but we would like to be able to do it abroad.
Yes, absolutely. But we have ovarian cancer as well in Chile, which is
that's right. You're right. Ovarian. Sorry.
Okay, I think we should wrap it up here. We're just 5 minutes over time, but I think we did very well to keep this on schedule. And Dr. Armisen can now go and cook for his children. That's very, very important. I have to do the same. Dr. Corvalán, would you like to say a few words to finish the session?
No, thanks to everybody. For this first session, we have this will be a 3 weeks meeting on Tuesdays between 8 to 1 p.m. So I would like to invite you for the next Tuesday when we have new speakers, and they will be for almost all this month of November. So everybody's invited, and invite your friends to join more people.
Okay, thank you for everybody for participating, and we'll see you next week. Goodbye.
Thank you very much.
Okay.
Bye-bye.
Bye.
Thank you. Bye.